Hello everyone. Good evening for the today's biology seminar, and this is the second seminar in this seminar series. And tomorrow we'll be doing chemistry seminar, same time from 8 p.m. till midnight. And last Wednesday we did a physics seminar. So this seminar series, the main objective to improve your knowledge and to improve your grades or the marks. And we have already shared with you WhatsApp group the two papers. One paper, Sri Javadpur Zone Support Seminar Paper Biology, and second paper by today's the guest lecturer for the seminar by Sir Channa Tharanga. So he'll be conducting part of the seminar today. And as you all know, biology section is very important in your oral science paper because you can easily get marks from this section. Especially you are getting around 12 to 13 biology MCQs out of 40 MCQs from both 10 and 11 syllabus. And structured essay, question number 1, compulsory question from biosphere lesson, that is grade 11 unit 15. And structured essay question number 2, also from biology, mainly from grade 10 work. Example, photosynthesis, the experiments, classification, reproduction lesson, inheritance lesson, and cells, structure of the cell, functions of the cell, tissues and all. And so essay type questions. Structured essay, you basically are getting out of 4 structured essays, out of 4 compulsory questions, you are getting 2 questions from biology, grade 11 unit 15, that is biosphere lesson, compulsory question number 1, and compulsory question number 2, that is also from biology. And essay types, essay type questions, you are getting question number 5 completely from biology, mainly from the system lesson. And you have already learned nearly 6 to 7 systems, reproductive system, and especially grade 11 chapter 6, respiratory, digestive, circulatory, blood circulatory, nervous system, endocrine system, from mainly from those systems, plus grade 10 classification, reproduction lesson, and inheritance lesson, also important for the essay type question number 5. And essay type, again you are getting biology, part of the biology, part of the question from biology grade, that is essay type question number 8, part A from biology, question number 8 part A biology, question number 5 also from biology essay type. So basically you are getting altogether nearly 4 essay type questions, or 2 structured and 2 essays, but not completely, question number 8 part A only. So therefore, to go for A pass, so biology part very important, because you can easily get marks, because no calculations, no equations, no balancing, no units involved, nothing much. So therefore there's less chance to lose marks from the biology section than from chemistry and physics. So therefore you have to target those biology essay type questions, you have to target full marks. And if you are good in biology, you can start with question number 5 essay type, and then question number 8 part A essay type. And structured essay anyway is compulsory, question number 1 and question number 2 biology. So without wasting much time, I'll start with now. Today there will be 2 sessions. So the first session I'll be conducting essay type questions, some selected essay type question from the support seminar paper from Sri Javadpur Educational Zone. And after that I'll invite Sir Channa Tharanga to continue the rest of the seminar, and that is the most vital part, and he'll guide you. And he is advanced level physics biology lecturer, advanced level English medium biology lecturer, and he has been conducting help most of the oral students last few years, oral badges also. And he can get the maximum out of you can get the maximum out of him today. and you can cooperate with him and get the maximum benefit. So I'll share the screen now. Today's there are two papers. First, I'll start the essay type questions from the support seminar paper, and then we'll continue the rest. You can continue the rest with Search and Taranga. Right. So you all can see the screen now. This is structured essay type question. You might get under oral paper the first question compulsory from the biosphere lesson. Anyway, the first question mainly targeting the ecosystem plus environmental issues in the world and energy crisis. So you all know even in our country the current one of the main current issues energy crisis. So therefore, it's environmental pollution, energy crisis, and ecosystem-based questions you can get, and diseases-related questions. So non-contagious or non-communicable diseases also they can ask under this question. I'll quickly go through this and you have to thoroughly study the biosphere lesson to get full marks for this question. But most of the things like common sense, you can think and write the answers even from grade 6 to 9 science knowledge, your previous knowledge also you can use. To write a food chain with 4 links that you find in this first. You can write, there are many alternative answers for this. You can write, you can see any food chain start with a primary producer. Any food chain start with a primary producer. You can start with plant material or grass or even any other plant you can write. So there's no definite answer for this. Actually, you can write any food chain with 4 links. So at the same time, you can put the question number and no need to write the question. If you have not got a hard copy, you can just write support seminar biology from Sri Javadhanpur Education Zone and essay type question number 1. A food chain with 4 links that you find in this forest. You can write, so you can write grass, and you can write here deer, and then you can write lion or tiger. I think you can write tiger here, or fox even you can write. Anyway, you don't get very colorful pictures of the diagrams in the oral paper, everything black and white, and then you can write say eagle or hawk. So you can write a simple food chain like, if you want to get start with, anyway the number 1 it should begins with a primary producer, and after that primary consumer. So we call this under tropic level. This is the first tropic level, this is the second tropic level, this is the third tropic level, and this is the fourth tropic level. So what are these called? Tropic level. This is one method of labeling the food chain, tropic levels. Or the other one you can write, the other method of naming this, you can write primary producer, primary consumer. So the grass you can write primary producer. Deer you can write primary consumer. Tiger you can write secondary consumer. And eagle you can write tertiary consumer. So that way also you can label this. Right. So in the exam they can ask even MCQs from this. So grass you can write primary producer. Deer you can write primary consumer or herbivore. Primary consumer, or you can write herbivore. Tiger you can write tiger or lion, or you can write within bracket herbivore. Then tiger you can write secondary consumer. So secondary consumer, but the tropic level is third. That's very important. So you can't tell secondary consumer and second tropic level. That is wrong. So tiger is secondary consumer and it is third tropic level. Eagle, that is tertiary consumer. Tertiary consumer. So this is the top carnivore. And one more thing you can write, along the food chain, number of organisms decrease. That was, if you don't know anything, it's up to you to decide. So if you don't know, you should better mark that in your short notebook separately. Along the food chain, number of organisms decrease. So based on that, we can construct number pyramid. Right? Number of organisms decreases. Number two, along the food chain, energy also decreases. So it takes the shape of a pyramid. Energy decreases. And number three, dry mass of organism. So we call it biomass. That also decreases. Out of this, one pyramid is always upright, that is energy pyramid. Energy pyramid, energy also decreases along the food chain. It's always upright, that's very important. It's always upright or it's erect. Always. But number pyramids and the biomass pyramid or pyramid of biomass, it can get inverted. They can get inverted. And next point, important point, energy flows only in one direction. In an ecosystem, what is the main source of energy? Sun. What is the main source of energy for an ecosystem? That is the sun. So it flows only in one direction, unidirectional. And in each tropic level, 90% of energy is dissipated. Around 90% of energy is dissipated due to the respiration, due to the metabolic activities, due to the growth, due to the excretion, due to the death of organism. So therefore, what is the amount flowing to the next level? Around 10%. From one tropic level to the next, only 10% of energy is entering. The rest is dissipated. We call it dissipated energy. So if you get a question related to this from food chain or food web, definitely they'll question about these things. What is the percentage of the dissipated energy and due to what? Due to which reasons, or what are the reasons for this energy dissipations? And this is a fixed ratio from previous level to the next level, always 10%. So example, if grasses, all the grasses in the grassland fix 10,000 kilojoules, what are the available for the deer, all the herbivores? 10% of that will be 1000 kilojoules. From 1000 kilojoules, 10% goes to the secondary consumers, that will be 100 kilojoules. From 100 kilojoules, only 10% goes to the last tropic level, that is 10 kilojoule. That means energy decreases along the food chain. So this is very, very important. Now all in one, because you don't have this in your textbook, but you can easily understand how energy flows along the food chain. And next one, a food chain is limited to maximum 4 or 5 links. Maximum 4 or 5 links due to this energy dissipation. You can't draw a food chain with 10-20 links. Maximum 4-5 links, because no energy available for the top carnivores. You can see if you put one more link here, whatever, it become 1 kilojoule. If you connect one more link to this food chain after eagle, then 10% into 10 kilojoules will be 1 kilojoule. So that is, energy is not sufficient to increase their population. Therefore, and the other most important thing, along the food chain, biomagnification take place. That means toxic chemicals, pollutants get accumulated, whose body will contain highest concentration of toxic chemicals, top carnivore eagle. So that was the highest concentration of toxic chemicals. So we call them bioaccumulating substances, get accumulated maximum in the top carnivore's body. That means herbivores contain less. That is called biomagnification or bioaccumulation. So there are substances that are getting bio. So I told everything related to this food chain now, and you can construct 3 ecological pyramids based on this, and you should know how to label the food chain based on the tropic level, first tropic level, second, third and fourth tropic level, or you should know how to label this in the form of primary producers, primary consumer, secondary consumer or tertiary consumer, and you should know If they give in the middle any link the total energy fix, then you should know how to predict the others either side. Right, I'll erase the screen. If you want, you can take a screenshot, or you can even request the recording after the seminar. Now let's see the questions. First one I have done for you. Number 2, write an example for each of the following types of organism from the above chain that you have written. Okay, let's see. First one, we predict the food chain grass. So you can write the producer, you can write grass. So in this food chain producer, you can write grasses. Secondary consumer, you can write primary consumer, deer. Secondary consumer will, now you have to write the answer based on the food chain that you have written here. You can write the others. So therefore you can write primary consumer deer, secondary consumer will be tiger. So the answer for B, secondary consumer, you can write tiger, producer you can write grass. Next one, draw and label energy pyramid for the food chain. So you can draw energy pyramid like this, but when you are drawing the energy pyramid, you have to label each level. You have to label. There are 4 links here. You can draw like this. So you can write separately. You can write grass, deer, tiger, and eagle. But you have to label this: first trophic level, second trophic level, third trophic level, and fourth trophic level. Or you have to write producer, primary consumer, secondary consumer, tertiary consumer, like that. You have to label this, otherwise you will not get full marks. Next one, explain why does the above pyramid is always upright. So energy pyramid will never become upside down. Energy pyramid is always upright. The reason, due to the flow of energy only in one direction. So you can write energy flows in an ecosystem only along the food chain. Energy flows along the food chain only in one direction. Energy flows only in one direction. Only in one direction, or unidirectional. That is the reason. Along the food chain, energy flows only in one direction. So we have to be very careful. Draw and label. If you just draw without labeling, you will not get. So therefore my advice, when you are answering the questions, target full marks, read the question twice, and pay attention to the question word and the active words of the question. Right, I'll erase the screen now. Sometimes there are students those who are going for A pass, but they lose more marks from the biology than from chemistry and physics because of careless mistakes. Explain why the above pyramid, that's done. Part 5. Consuming plant products by man is the solution for energy crisis. So even in the future you will have to get adapted to the seafood also, seaweed. Explain the above statement using energy pyramid. So you can write here, producers contain more energy than in the consumers. In an ecosystem, producers fix energy. In an ecosystem, who fix energy first? Producers, primary producers fix energy first by photosynthesis. That's the reason. And total amount of energy in producers more than in consumers, right? The total amount of energy in an ecosystem in producers greater than in consumers. Consumers means carnivores, herbivores, and omnivores. If you get the total energy in all the animals, it's less than the total energy in plants. That's the reason. Name 2 elements that circulate in the above forest. This time O-level paper, this you have to pay attention to these 2 cycles: carbon cycle and nitrogen cycle. Carbon cycle and nitrogen cycle. Sometimes you can get essay or structure essay. 2 elements that circulate, you can write 1 element, you can write carbon, or you can write even the symbol, and the other one in your syllabus, nitrogen, N. That's why, in other than that, we can talk about sulfur cycle, oxygen cycle, water cycle, hydrological cycle. So these are elemental cycle, geochemical cycle. B. Greenhouse effect. Definitely you will get questions related to the environmental issues. Acid rain. Due to air pollution, you get acid rain, ozone layer depletion, photochemical smog, and global warming. Acid rain, ozone layer depletion, photochemical smog, and global warming due to air pollution. So you should know under any environmental issue, you should know the pollutant. Number one, you should know the pollutant. Number two, you should know the pollution. Number three, you should know from where these pollutants are released to the environment. And number four, you should know harmful effects. And number five, you should know how to minimize. Again, I'll repeat. Under any environmental issue, whether it's acid rain or global warming or ozone layer depletion or land pollution or water pollution, you should know the pollutant. And you should know the pollution, how that happens, how that pollutant causes the pollution. And you should know sources of the pollutant, from where it is coming, sources of the pollutant. And you should know the harmful effects and method of minimizing. So under any environmental issue, they can ask, you will get the questions under these five, not anything out of that. Pollutant, it can be gaseous pollutant or liquid or solid pollutant. Pollution, you should know how that happens. And from where it is released, sources, and what are the harmful effects and method of minimizing. Example, acid rain. What are the pollutants? Example, sulfur dioxide, nitrogen dioxide. Pollution, you should know, acidic gases dissolved in the rain and reduces the pH value of water below 4.6. Then sources, from where these acidic gases released to the environment, from the atmosphere. And harmful effects of acid rain, dissolving of minerals, increases the corrosion of valuable properties, and method of minimizing. So all these you should know. And just one example. So now B1, name two types of greenhouse gases. This is very, very important. Remember, greenhouse gases means polyatomic gases. Monoatomic and diatomic gases are not considered as greenhouse gases. What are greenhouse gases? Polyatomic gases with a you have learned about this polar molecule and non-polar molecule. Polyatomic gases can act as greenhouse gases. Any polyatomic gas. If you want, you can write separately. Monoatomic. Example, helium, neon, argon. And homodiatomic. That means nitrogen, oxygen, hydrogen are not greenhouse gases. Monoatomic and homodiatomic gases are not greenhouse gases, are not considered as greenhouse. Please write it, because MCQs they can ask this question. Are not considered as greenhouse gases. Otherwise, nitrogen in the atmosphere 78%. If it is a greenhouse gas, we are in a big trouble. Nitrogen 78%, oxygen 21%, helium not. They are IR inactive. Greenhouse gases are IR active. They can absorb IR, infrared, IR active. Greenhouse gases are IR active. One of the main greenhouse gas, carbon dioxide. Carbon dioxide can absorb infrared, IR. Other than that, you can write CFC, chlorofluorocarbon. Other than that, you can write CH for methane, and even water vapor. If someone write sulfur dioxide and nitrogen dioxide also, you can consider that as greenhouse gases, but the greenhouse potential by SO2 and NO2 less, because they are acidic gases, they get washed away with the rain. So in your syllabus, there are four main greenhouse gases: carbon dioxide, chlorofluorocarbon CFC, water vapor, and CH for methane. Now CFC, a greenhouse gas as well as ozone depleting gas. CFC, please write, CFC causes both the issues. It's a greenhouse gas as well as ozone depleting gas, chlorofluorocarbon or halogenated hydrocarbons. So they are asking only two types of greenhouse gas. Other than that, you should know the gases which cause acid rain. There are two gases, sulfur dioxide and nitrogen dioxide cause acid rain, and sulfur trioxide also cause acid rain. These are the three gases which cause acid rain. And there are two gases which cause ozone layer depletion. So this is acid rain. And what are the two gases which cause ozone layer depletion? CFC and nitric oxide. These two causes ozone layer depletion. Ozone layer depletion. So mainly you get three environmental. Number one, greenhouse effect or global warming. Number two, acid rain. Number three, ozone layer depletion. Number four, photochemical smoke. Photochemical smoke causes by oxides of nitrogen. I put NOx, oxides of nitrogen and hydrocarbons. I put CH. What are oxides of nitrogen and hydrocarbons causes photochemical smoke. So you can definitely get at least a MCQ from this if you don't get any essay or structured essay. Right. Remember, not only the answers for this question, you should think about the other related questions they could ask under this. Right. Next one, mention the ways by which the above gases release to the environment. Combustion of coal, carbon dioxide. So you have to be careful because it depends on the gas that you write for the part 1. If you write carbon dioxide, you have to write the sources of that. So you can write carbon dioxide, combustion of fuel. Combustion of fuel. Or combustion of coal. In Sri Lanka, to generate electricity, we burn large quantity of coal, diesel. So then it causes the emission of CO2 gas. In addition to that, you can write CFC from refrigerators, air conditioners. Right? Two indirect effect of emitting greenhouse gases. Now there are direct effect and indirect effect of environmental. They have given this in chapter 15. You have to be very careful. You should, if you want, I'll tell the page also. Direct effects of environmental pollution and indirect effect. So direct effect means acid rain, global warming, ozone layer depletion, eutrophication. Those are direct effect. What are the direct effect of environmental issues? Eutrophication, acid rain, global warming, biomagnification, acid rain, ozone layer depletion. So indirect effect means loss of habitat, reduction of crop yield, desertification. So these are the indirect effect. Due to the global warming, you can get desertification. Formation of desert can increase. Crop yield can decrease. It can lead to food crisis. Right? Loss of habitat and desertification. Next one, write 2 steps that can be reduced greenhouse effect. So use of renewable energy sources instead of coal and petroleum fuel. Use of renewable energy sources instead of petroleum fuel. Or you can write reforestation. Reforestation also a solution for this. Growing more plants. Reforestation. So that's about this environment. Other than that, they can ask about this is also very important. Greenhouse effect is, most of the students think greenhouse effect is equal to global warming. That is completely wrong. Greenhouse effect is useful process. Without greenhouse effect, the earth would be a cold planet. Temperature will be minus. Actually, the problem is enhanced greenhouse effect. If greenhouse effect increases beyond the limit, we call it global warming. Therefore, global warming is due to greenhouse effect. Actually, global warming is due to the enhanced greenhouse effect. Global warming is due to enhanced greenhouse effect, or greenhouse effect increasing beyond the permissible level. Without greenhouse effect, temperature will be very low in the earth and it will not support the life. Therefore, without greenhouse gases also we cannot survive in the earth. Increase in of greenhouse effect also problem. decreasing of greenhouse effect also problem. Therefore, greenhouse effect is a very useful process which helps to regulate the temperature of the earth. Now what we experience, global warming, enhanced greenhouse effect. But opposite of this, global cooling. If greenhouse gases decreases, then we call it global cooling. So both not good. You can't label CO2 as a bad gas. Without CO2, no photosynthesis. Without CO2, no food production in the plant. Without CO2, no greenhouse effect decrease. Then it can cause global cooling. Therefore, CO2 also equally important as oxygen for your respiration. So don't forget about that. And large quantity of CO2 retains in water. More than 70% of the earth covered by water. So CO2 available in the dissolved form. Ocean carbon sink. Next one. CKD, chronic kidney disease. This also they have given in your grade 11 textbook, chapter 15 about CKD. Remember this time all over this respiratory system and excretory system bit important. Respiratory and excretory, especially respiratory system. At the end of this essay type question, there's a question from respiratory system also. And CKD or chronic kidney disease, that means it cannot be reversed. It's a gradual loss of the kidney functioning, mostly in agricultural areas in Sri Lanka. Right, two factors that have been identified for causing CKD, chronic kidney disease. So one of the main reason use of agrochemicals. One of the main reason for spreading this CKD in those areas due to the exposure to agrochemicals, heavy metals. Agrochemicals. That's why in the agricultural areas, exposure to heavy metals, contact with agrochemicals and heavy metals. What are heavy metals? Zinc, mercury, arsenic, cadmium. Other than that, there are many other reasons. So drinking water with high concentration of fluoride and the other chemicals, use of drugs without any control, consumption of liquor, dehydration. There are many other reasons, but mainly agrochemicals, and agrochemicals you can write mainly. Two factors they ask. Next one, two symptoms of the above disease. CKD. One of the main symptom, you can the no urine output or little or no urine output, and swelling of feet. Swelling of feet because the toxic substances, urea, uric acid, those things get accumulated in blood. Swelling of feet. Presence of protein in urine. Presence of protein in urine. Your urine should not contain any protein. But CKD patients can contain this. In addition to that, rashes on the palm and soles. Rashes on the palm and the soles. So any of these you can write, you can refer page number 198, 99 in your grade 11 textbook. Page number 199, 98, 198, 99 in your grade 11 textbook, last lesson, biosphere lesson. Next one, mention two examples for the sustainable agricultural uses to avoid CKD. Here it should be avoid CKD. So one is multi-crop agriculture. Multi-crop agriculture, multi-cropping. Then crop rotation. Then use of organic fertilizers. Use of organic fertilizers. Use of organic fertilizers. Crop rotation. Multi-cropping means growing different types of crops in the same land at the same time. Crop rotation means instead of growing the same crop again and again in the same land, you rotate it. Next season you grow some other crop in that area. and use of organic fertilizers instead of, and biological pest control. Biological pest control. You can write any two of this. That is question number 1. So therefore, anyway, you have to study before you study in any other biology lesson. You have to study grade 11 biosphere lesson first, because you are getting the compulsory question. There's no choice. You have to answer that question. Structure say question number 1, and you have to study these things also other than this, especially food mile. Please pay attention to this food mile, water footprint. Definitely you can get questions from these areas. Water footprint, carbon footprint, eutrophication. Other than this ecosystem, population and ecological pyramids, environmental pyramids, they can ask questions from food mile, water footprint, carbon footprint, eutrophication, carbon cycle and nitrogen cycle. With carbon cycle, they can relate the global warming also, nitrogen cycle. Those are the other areas you have to pay attention. So therefore, definitely you can get full marks this question number 1, because it's not hard, no calculation, no balancing, no equations, no units. So just to study, and you can get definitely full marks. Right, question number 2. Before I start question number 2 now, I would like to invite Sir Channa Taranga to continue the rest of the other paper, and after that, if there's time left, I'll come back to this paper, because I think today actually the guest speaker for this biology seminar, I invited him without wasting his time. Also, I thought of inviting him now, and you can ask any question you have, any doubts related to that, because he is very very popular and very well renowned this biology A-level English medium biology lecturer, and he is a lecturer of a private university also at A-level English medium, and even you can join with him for a advanced level local syllabus and Cambridge EdExcel even biology he is conducting, and he has taught many students past few decades, and there are many undergraduates and those who have passed out from the universities, his past students, and today he will guide you, and you can get maximum out of him today. So I would like to invite Mr. Channa Taranga to continue the rest of the seminar.
Thank you, Jagat sir. So, okay guys, so we will start with the MCQ paper. So Jagat sir gave an introduction. So my name is Channa Taranga. So I'm currently working as a lecturer in microbiology and immunology, senior lecturer in microbiology and immunology, and also a consultant in academic affairs in Helixion Innovation. It's a new innovation company. So we will quickly move into the biology MCQ paper. Okay. So give me one moment. Okay. So now if you look at, guys, now the thing is it is very easy to score marks for biology MCQs. Okay, but only thing is you have to be very careful, especially when you read the questions. It's very, very important. Okay, you have to be very careful. So if you can't understand, just read it twice or thrice. That's okay. Biology questions are very easy to answer. But only thing is, most of the times, our students, what they do is they don't read it properly. Then they, pick something, some wrong answer. So if you look at the first question, guys, now what is the macromolecule without any sugar component? Now, if you look at this question, here you have two words that you need to pay attention. These two words, macromolecule and sugar component. Now, in this question, we are asking what is the macromolecule without any sugar component. So, out of these answers, whatever the answer that you are going to pick should be a macromolecule. The other thing is it should not have any sugar component. What did you pick? If you want, you can share your answer with me. You know, you can just send it through the chat. So if you look at glycogen, guys, glycogen is a macromolecule, but also it has a sugar component, right? I'm getting some answers, yes, but third one answer is the correct one. If you look at the glycogen, Putala, glycogen contains glucose. So definitely, even though it is a macromolecule, it has a sugar component. It is made up of glucose. Then someone told me RNA, but RNA is not the answer. Why? RNA is a macromolecule, but then you also need to recall what is the now RNA is composed of nucleotide, right? The monomer of the RNA is ribonucleotide. Now we know that ribonucleotide is something like this, right? So you can see here there is a nitrogen base. Now here you have phosphate group. So you have a nitrogen base and you have phosphate group. And then this one, what is this? Pentagon. This is the pentose sugar. So in RNA, in ribonucleotide, you have ribose. In DNA, you have deoxyribose. This is a sugar, okay? It's a sugar component. It's a carbohydrate. So RNA is not the answer. Now, I saw a few answers saying phospholipids. Again, yes, it doesn't have any sugar components. That's true. Perfectly true. But as I told you, this question carries two important words, macromolecule and sugar component, without any sugar component. So when you say without any sugar component, phospholipid should be correct. But is it a macromolecule? Is it a polymer? No. Guys, now you have learned that there are 4 main biomolecules, right? Carbohydrates, or we call it hydrates of carbon, carbohydrates, lipids, proteins, and nucleic acid. Now, if you look at carbohydrates, there are 3 types of carbohydrates, no? Right? You have monosaccharides, disaccharides, and polysaccharides. Out of monosaccharides and disaccharides, they are not macromolecules, but polysaccharides are macromolecule. Macromolecule means made up of large number of monomers. Actually, usually the mass sometimes can go beyond 1000 kilodaltons. So then if you look at proteins, protein is a macromolecule because protein is made up of collection of amino acids. So protein is a polymer and the monomer is the amino acid. Nucleic acid also a polymer, right? It's a macromolecule. Lipids, Is it a macromolecule? No. Why? Lipid is consist of glycerol and fatty acids. Just one glycerol molecule like this. One glycerol molecule. This is a glycerol molecule. Huh? And then there are 3 fatty acids connected to the glycerol through the ester bonds. Fatty acids. Okay. So it's not a macromolecule. It's not a macromolecule. There are 3 fatty acids and 1 glycerol. So just the 4 molecules, right? Combination of 4 molecules. I mean, combination of 4 molecules cannot be considered as a macromolecule. No? So lipids or the phospholipids cannot be considered as a macromolecule. Even though it doesn't have a sugar component, it's not a macromolecule. So what is the answer? Albumin, which is a protein. Okay. Glycogen is not the answer because even though it is a macromolecule, it has sugar component, which is glucose. RNA is not the answer because even though it's a macromolecule, it has ribose in it. And ribose is a sugar. Phospholipids doesn't have any sugar component, but it's not a macromolecule. So the answer is albumin. Hope you understood, right? If you have any questions, Putala, just raise your hand, you can just put it in the chat. If I have time, I will answer it. If you are very and if there is a big problem, then just raise your hand and I will unmute you. You can just ask any questions. Okay. Look at the second question. Deficiency of which mineral causes yellow or brown patches in leaves? Now the answer is actually potassium. So deficiency of which mineral causes yellow or brown patches in leaves? The answer is potassium. Okay. Now here, guys, the second question, now you will definitely get something from minerals or vitamin. So you have to study that. Okay, because there is no short ways of, memorizing them. So you have to study that part. So, and if you look at the third one, an organelle not containing any DNA inside it. Now, all these things are in your book. Okay, if you actually look now, sometimes you don't get all the MCQs like this for biology paper. Okay. But then when I make the paper, I made it a bit harder because, I made everything like analytical questions. There are few analytical questions, but not all. Most of the times, it can be direct questions plus some, you need to do some analysis. So, but only thing is, you have to be very thorough with your biology syllabus and study it properly. If you know these things you can very easily answer these questions. Now look at the third question. An organelle not containing any DNA inside it. This is very easy. Now nucleus, definitely it has DNA, right? So obviously it's not the answer. Chloroplast and mitochondria. You guys should know chloroplast and mitochondria contains DNA inside that. Chloroplast and mitochondria contains DNA inside that. However, Golgi body doesn't have any DNA. Golgi body doesn't have any DNA. Okay. Nucleus, mitochondria, and chloroplast contains DNA, but not in Golgi bodies. So, if you look at only nucleus, mitochondria, and chloroplast contains DNA. If you take a particular cell. If it is a plant cell, then both nucleus, chloroplast, and mitochondria contains DNA. In animal cell, nucleus and mitochondria contains DNA. Now the fourth question. A cell has 16 pairs of chromosomes. At the end of the cell division 1 of the meiosis, at the end of the division 1 of meiosis, how many chromosomes will there be inside the nucleus of one daughter cell? Now, again, this question also, reading is very important. Reading is very important because I know most of you guys might have put 8 as the answer. I'm pretty sure. Okay? Because you don't read properly. And I'm asking about the meiosis division 1. Look at here. A cell has 16 pairs of chromosomes. 16 pairs of chromosomes. Right? Yeah, one student gave me correct answer. So 16 pairs of chromosomes. That means how many chromosomes? 32. 16 pairs means 32. 32 chromosomes. So definitely in the meiosis division 1, this will be half, right? So if you have 32 chromosomes, after the division 1, the 2 daughter cells will receive 16 each. You have 32 chromosomes, there are 16 pairs. Each daughter cell will receive 1 chromosome from each pair. So if you have 16 pairs, each daughter cell will receive 16 chromosomes. 16 pairs means 32 chromosomes. From each pair, 1 daughter cell receive 1 chromosome. Understood? So you have 16 pairs of chromosomes, that means totally 32. In meiosis 1, it would be half. So daughter cells will receive only 16 chromosomes each. Hope you guys are clear. The fifth one, an organism without cellular organization. An organism without cellular organization. Now, if you look at Chlamydomonas, Chlamydomonas is a algae, the unicellular algae, or you can call them protophyta. It belongs to the kingdom Protista, and it's an eukaryote. Okay, it's an eukaryotic cellular organization. It has the eukaryotic cellular organization. So no problem, Chlamydomonas has eukaryotic cellular organization. There are 2 cellular organizations. Now, what are the 2 cellular organizations? In organisms, we can observe 2 types of cellular organization. What are the 2 types of cellular organizations that we can see? Prokaryotes, prokaryotic organization, and eukaryotic organization. Eukaryotic organization. Now, prokaryotic cellular organization can be seen in two domains, domain bacteria and domain archaea. Domain bacteria and domain archaea. While eukaryotic cellular organization can be seen in domain eukarya. Now, Chlamydomonas is a protista, belongs to the kingdom Protista, which is an eukaryotic organism. Amoeba is a protozoan that also comes under Protista kingdom. So it's also an eukaryotic cellular organization. E. coli bacteria, a prokaryotic cellular organization. Guys, remember, this viruses doesn't have any cellular organization. Viruses doesn't have any cellular organization. They don't have any cellular organization. Because, you know, that there is no cell. You can't tell whether it is living or non-living. Okay, so the answer is Ebola virus. Ebola virus. Select the false statement. Organisms of domain bacteria are sensitive to antibiotics. That's correct, right? Bacteria are sensitive to antibiotics. So who are not sensitive? Domain Eukarya and domain Archaea. They are not sensitive. But bacteria are sensitive to antibiotics. Bacteriophage does not belong to any domain. Bacteriophage does not belong to any domain. That is correct. Bacteriophage is a virus, right? It's in your book. Bacteriophage is a virus. Virus that infect bacteria, that eat bacteria. That is why we call it bacteriophage. Something like this. I think you might have seen the picture also. Bacteriophage is something like this, you know. If you have watched Matrix movie, they have represented the computer viruses also like this. So this is a bacteriophage. Okay. So bacteriophage does not belong to any domain. That's correct. Okay. Now in this question, actually there are two answers. Because if you look at the third one, domain Archaea is evolutionary closer to domain bacteria than domain Eukarya. Now, according to this statement, domain Archaea is evolutionary closer to domain bacteria than domain Eukarya. That is wrong. A lot of people think this is correct, but this statement is false statement. If you look at the evolutionary tree in your book, you can see it. It's like this. Bacteria. Then you can see there is a common ancestor between Archaea and Eukarya. Even though Archaea also a prokaryote, and earlier we consider Archaea as a part of bacteria because they are prokaryote. However, even though Archaea are, you know, like bacteria, because Archaea is a prokaryotic organism, they don't have a proper nucleus. Even though it shares those similarities with bacteria, actually Archaea is more closer to Eukarya in evolutionary wise. Then if you look at the fourth one, Nephrolophis is a non-flowering seedless plant. That's correct. Non-flowering seedless plant, that part is correct. With a thalloid shaped body, that's not exactly correct. Because if you look at the Nephrolophis, it's, you know, ferns. Ferns is not like thalloid, no? Okay. So you can consider, and in this level, you can consider third and fourth both are false. Then seventh one, an animal without a coelom. You know what is a coelom, right? Coelom is a body cavity surrounded by the mesoderm. So centipede is an arthropoda. It has a coelom. Guys, remember, from the phyla that you have studied, only Cnidarians doesn't have a coelom. So here the answer is jellyfish. The centipede, snail, as well as Nereis. Nereis belongs to the kingdom, phylum Annelida. Snail belongs to the mollusca. Centipede belongs to the arthropoda. So the answer is jellyfish. Okay. So one student asking, so for the sixth question, whether third and fourth both are can be considered as the correct answer. Yes, because third and fourth both are false. You can consider both the answers are as correct because third and fourth both are false. Both are false statements. So the seventh question, the answer is, the seventh question answer is jellyfish. Okay, if you look at the eighth one, a plant that uses both bulbils and suckers for natural vegetative reproduction. So a plant that uses both bulbils and suckers for natural vegetative reproduction. The answer is pineapple. You know, breadfruit, they use their roots, paddy and jute. So the answer is pineapple. Pineapple is the one which uses both bulbils and suckers. So this part also bit important, guys. So just read these examples for different vegetative, you know, there are underground stems and all these things. So just, you know, study that part and go ahead. And one student asking me about the coelom or coelenteron. Coelenteron is a body cavity, you know, surrounded by our mesoderm. There are 3 germinal layers, no? If you remember, diploblast and triploblast, right? The jellyfish is, or the cnidarians are diploblast. And the other organisms like nematodes or Annelida, Arthropoda, they are triploblast. Triploblast means there are 3 germinal layers, endoderm, mesoderm and ectoderm, like this. So cavity produced completely surrounded by mesoderm. The middle germinal layer is known as the coelom. So all our organs, you know, all our body organs are located in the coelom. If you look at a human also, all the organs are located in the coelom. Okay. It's a secondary body cavity, but you don't have to know all these things because it's not that important. Okay, there are some other questions. Give me 1 minute. Now, some of you guys have asked me why the about the other answers, right? So if you look at Putala, now what can you say about the breadfruit? Now, what are the different vegetative reproduction methods? Now, I will just quickly go through that. What is meant by the vegetative reproduction? Vegetative reproduction means you are using a vegetative part, you know, part that we use for the growth, for the reproduction, usually. Okay. So if you look at now, there are so many vegetative parts that can be used in reproduction, right? So you can use roots. So examples would be, you know, like breadfruit. Now here the breadfruit, it's actually roots. Then if you look at the second answer, paddy. So well, like what is the vegetative reproduction method? I mean, these plants are completely capable of doing the you know, usual the sexual reproduction also, not only asexual reproduction, but then they also have vegetative reproduction. So if you look at paddy, now they use suckers. Now, but my question is bulbils and suckers both. So even though paddy uses suckers, it doesn't use bulbil, right? So that is why we can't take paddy as the answer. We can't take breadfruit as the answer because it uses roots. We can't take paddy as the answer because it only uses suckers. But if you look at pineapple uses suckers, and also pineapple uses bulbils. Okay. However, if you look at jute, it only uses bulbils. Understood? So, and one student asking me about the COVID-19 virus, whether any virus doesn't have a cellular organization, Pudala. Okay. Whether it is COVID-19 or whatever, it doesn't have cellular organization. Okay. Jute is a plant that we use to get the fibers. So, if you move into the other question, so the eighth one, the answer is pineapple, because it uses both bulbils as well as suckers. Now, if you look at the question 9, a non-protein hormone involved in menstrual cycle. Again, this is also a question that you need to analyze a little bit more. Okay, this is a question that you need to analyze a little bit more. Now, here also, there are two terms that you need to look at. Now, I'm getting a lot of answers, but I know most of you have selected the wrong answer. So, non-protein hormone is the one term that you need to look at. And the other one is menstrual cycle. It should be a non-protein hormone, and also it should be involved in menstrual cycle. Now, FSH and LH both are involved in the menstrual cycle. Okay. However, both FSH and LH are protein hormones. It's made up of proteins. Thyroxine is not involved in the menstrual cycle, and it's also, a protein hormone. But if you look at the progesterone, it's involved in the menstrual cycle, and also progesterone is not made up of proteins. It's a steroid hormone. Progesterone. It's not made up of proteins. FSH stands for follicular stimulating hormone. Luteinizing hormone is LH. Both are secreted by the pituitary gland. It's in your syllabus, the 6th lesson. Okay. Then if you look at the 10th one, a blood-related disorder caused by a gene on an autosomal chromosome. A blood-related disorder caused by a gene on an autosomal chromosome. Now, this is also very important. There are two terms that you need to pay attention. So, one thing is blood-related disorder. So, this disorder should be related to blood. Okay. So, blood-related disorder, and it also in the autosomal chromosomes. Now, if you look at hemophilia, yes, it's a blood-related disorder. You know, there is a problem in the blood clotting when you have factor X actually. So, there is a problem in the blood clotting factor. So blood won't clot properly. However, hemophilia gene is not located on an autosomal chromosome. Now, see guys, we have how many chromosomes, chromosomal pairs? We have 46 chromosomes and 23 pairs, right? Out of these 23 pairs, 22 chromosomes are known as autosomal chromosomal pairs. Then we have 6 chromosomes. So X and Y. Females, they have 2 X chromosomes. X chromosomes are large, okay? And males have 1 X chromosome and one Y chromosome. Since the X chromosome is large, apart from the sexual characteristics, X chromosomes has other genes as well. So the gene for hemophilia is located on the X chromosome. So we cannot consider hemophilia as the answer, because even though it's related to blood, it's not on autosomal chromosome, it's on a sex chromosome, X. Then color blindness. Color blindness is not related to blood, and also it's located on sex chromosomes again, similar to hemophilia. Albinism is located on autosomal chromosomes, but it's not related to blood. So our answer is thalassemia. Thalassemia gene is located on autosomal chromosome, and it's related to the blood. Actually, it's related to the hemoglobin production. So due to the mutated gene, there will be a problem in the hemoglobin protein. So the thalassemia is very, the high prevalence of thalassemia can be seen in the Kurunegala district. Okay. So you can see a high prevalence of thalassemia in the Kurunegala district. So that is why especially if you are from Kurunegala district, if you are going to marry one of your blood relatives, like the marriage between Nana Masina, like marriage between cousins, then definitely it's not a good idea. Or else, even if you are living in Kurunegala district, then definitely the people from Kurunegala, before getting married, it is better if they check whether they are thalassemia carriers, because the thalassemia disease gene is not dominant gene, it's a recessive gene. However, when 2 carriers meet, then it's like this. Now imagine you take capital T and simple T. Capital T is the healthy gene, simple T is the disease gene. So there are 2 marriage between 2 carriers. So what will happen? Now this is the genotype of the mother and the father. So these are the possible gametes. These are the possible gametes. So what will happen? You may get completely healthy person. You may get a carrier, another carrier, and there is a 1/4 chance that you are getting a thalassaemic patient. So that is why it is very important to check whether a person is having mutated gene for thalassemia before they get married, especially if they are in Kurunegala district, because of the high prevalence. Again, that is why it is people advise not to get married between the blood relatives. Not only thalassemia, other genetically inherited diseases also can go through the generations. So that is one of the main reason, this in Kingdom of Egypt, in Pharaoh, time of Pharaoh, they used to marry their sister. So little by little, all these bad genes got accumulated. Then when later on, almost all the kings and queens, they had genetic disorders. So that is one of the reasons which led to the destruction of the Egyptian empire. And that is why still in the European continent, most of the European royal families have this hemophilia gene. One person asking, is there any specific reason? Yes, Kurunegala district has a high prevalence of thalassemia. It's not in your syllabus. It's actually related to malaria, because Kurunegala district had a huge malaria outbreaks. Okay, a lot of people died due to malaria in Kurunegala district, like not recently, but long time back. So people with, you know, thalassemia mutated gene, especially the carriers, has some sort of a protection against malaria. So what happened was, due to the malaria, a lot of people died, but the people with the thalassemia gene, the carriers of the mutated gene, survived. So that is why now we have high prevalence of thalassemia mutated gene in Kurunegala district. Okay. If you go to the 11th one, a cell with a nucleus. Now you may think this is an easy question, but again, sieve tube elements are in the phloem tissue. They don't have a nucleus, right? They don't have a nucleus because in order to increase the efficiency of phloem translocation, in order to send the, you know, that food, the phloem, whatever the sugar solution through the cells, they don't have a nucleus, because if they have the nucleus, then and the other large organelles, it affects the flow of the sugar solution. So the sieve tube elements doesn't have a nucleus. Tracheids, no, it's a part of xylem tissue. RBC, no. RBC also red blood cells, especially the adult red blood cells, the mature red blood cells, they don't have a nucleus. So the answer is companion cells. Companion cells actually always stay with the sieve tube elements. Now, if you look at the sieve tube elements, the companion cells are like this. So the companion cell has a nucleus. So companion cell is the one which regulates all the functions of the sieve tube elements in the phloem tissue. So companion cells, they have nucleus. Select the false statement. There is no blood supply to the epithelial cells. That is correct. To the tissues, there are nerve supplies, but into the tissues, there are no blood supply into the epithelial tissue. The matrix of the blood tissue is not secreted by blood cells. That is also correct. Usually, guys, remember, blood tissue is a type of connective tissues, right? Now, connective tissues, they have a matrix and they have cells and they have fibers. There are three main components in connective tissues. They have a matrix, they have cells, they also have fibers. Usually their matrix is semi-solid. And then usually in the connective tissues, the matrix is secreted by the cells. If you look at even bones, our bones is a type of connective tissue, our cartilage. So all these cartilage matrix or the bone matrix are secreted by the cells in that connective tissue. So the cartilage matrix is secreted by cartilage cells. Okay. Then bone is secreted by osteocytes. However, in blood, that is different. Even though blood is a connective tissues, one thing is the matrix of the blood, that means blood plasma, is not secreted by blood cells. Blood plasma is not secreted by blood cells. The second difference between the normal connective tissues and the blood would be that in blood, under the normal conditions, there are no fibers. In connective tissues, there are fibers. But in blood, under the normal conditions, there are no fibers. However, when blood clotting occurs, there will be fibers. But under the normal conditions, there are no fibers. Then the third one, skeletal muscle fibers are uninucleate cells with striation. Now, this part is correct. However, the skeletal muscles are not uninucleated. They are multinucleated. nucleate cells. The fourth one is correct. Dendrites transmit impulses to the cell body of the neuron, right? If you look at this, dendrites transmit the impulse while the axon takes it out. Okay. So then what is the false statement? Third one. Skeletal muscle fibers are uninucleate cells with striatum. This part is wrong. Uninucleate. 13th, an internal factor essential for photosynthesis. Internal factor, this is very easy. Chlorophyll, right? Everything else, oxygen, carbon dioxide. Sorry, oxygen is actually not an essential factor. It's a product. Then if you look at carbon dioxide and water, they are factors required for photosynthesis, but they are not internal factors. So the internal factor is chlorophyll. Now the 14th question, look at this very carefully. When a pure breeding tall plant cross with pure breeding short plant, all the plants of the F2 generations were tall. F2 generation plants were crossed with pure breeding short plants again. What is the phenotype ratio of progeny plants? Okay, guys, I'm sharing the whiteboard. Hope you can see. Okay. Now imagine tall plants. True breeding tall plants and true breeding short plants. Homozygous tall and homozygous short. So if you take capital T as the tall the gene for the tall, then simple T is for the short. So always there are two factors, two genes. You call homozygous tall, homozygous short. So the gametes would be only one type, no, capital T, simple T. So when you cross them, you get capital T, simple T. 100% this capital T, simple T. They are tall. Okay. Now this is the F1 generation. This is the F1 generation. This is the F1 generation. Now what has happened? We have taken the F1 generation. That means this. And again we have let it to be crossed with true breeding short or homozygous short. These type of cross are known as test cross. These type of crossing is known as test crossing. Why do we do a test crossing? Now imagine we get a tall plant. Now when we get a tall plant can have two types of genotypes, right? Tall plant can be capital T, simple T, or capital T and capital T. Whether it is capital T and simple T or capital T and capital T, the phenotype is always tall, right? So when you get a tall plant, if you want to identify what is the genotype, in tall plants, there can be two genotypes. Capital T, simple T, capital T, capital T. So to identify whether it is capital T, simple T or capital T, we go for the test cross. So this is a test cross. So when you let the F1 generation to be crossed with the pure breeding short plants, what type of gametes you will get? Capital T, simple T. Here you have only one type of gamete, simple T. So you get capital T, simple T, or you can get simple T can go with simple T. There are two types of gametes here, one type of gamete here. So capital T can go with simple T, or simple T can go with simple T. So this is the genotype ratio, 1:1. Tall and short, this is tall, this is short, 1:1. Tall and short, 1:1. This is tall, this is short, 1:1. 1, 1. Ratio is 1:1. Hope you have understood. So the answer is second one, tall, short, 1:1. What is the sweetest sugar? This is a very direct question. The answer is fructose. Fructose also known as the fruit sugar. Okay. Out of these sugars, which has the least sweetness, out of these four, it's lactose. The lactose is not that sweet. The lowest sweetness out of all the sugars that we know, there are 3 monosaccharides, glucose, fructose. So fructose is the sweetest sugar. It is also known as the fruit sugar. Then galactose. So galactose, the sweetness is very, very low. Fructose has the highest sweetness. Then if you look at the disaccharide, now these 3 are monosaccharides. Then we have disaccharides, maltose, which is made by glucose and glucose, disaccharide. Sucrose, which is in the normal sugar that we eat, sugar cane. Sucrose contains glucose plus fructose. Lactose, also known as a milk sugar, consists of glucose and galactose. Glucose and galactose. Okay. So the fructose is the sweetest sugar out of all the sugars that we know. What is the blood vessel that directly connects 2 organs without connecting to the heart? Anyone knows the answer? What is a blood vessel that directly connects? This is the only blood vessel that connects 2 organs. Yes. The third one, hepatic portal vein. Hepatic portal vein, guys. Now, pulmonary vein is, there are 2 pulmonary veins, left pulmonary vein and the right pulmonary vein, coming from the lungs to the heart. So it connects lungs and the heart. Okay. Pulmonary veins. Renal artery, heart and the kidney. Heart and the kidney. Hepatic artery, heart and the liver. Heart and the liver. Hepatic portal vein is the one which connects liver and the small intestine. Once we eat, or absorption during the digestion process, the absorption will be completed in the small intestine, right? So once the small intestine absorb all the products of digestion, like sugar, vitamins, everything, amino acids, everything, then that will not directly be connected to the main blood supply. Everything in the small intestine will be first moved to the liver. Then only it will be connected to the main blood circuit. So the hepatic portal vein connects 2 organs. This is the only vein that connects 2 organs, only blood vessel that connects 2 organs. Actually, this happens why? Because what is the main function of our liver? Our main function of our liver is the detoxification. If you eat some toxins, it has to be detoxified. It is done through the liver cells. So that is why everything that we eat should go to the liver first. That is why always our liver is affected by the toxin. That is why even though we people like to eat, liver, right? Not the human liver, but the animal liver. If you are meat lover, you like to eat the liver, but it's not good. Okay, because if they have any toxins, all the toxins, I mean, it's good thing because it has a lot of nutrients, but the same time with the nutrients, it also can have lot of toxins in it. That is a problem. Liver contains a lot of nutrients, but same time it can have a lot of toxins. So during, autopsy, when people die, in order to check whether they have taken any toxins, whether they have been poisons or not, you usually check the liver. You take the liver, cut the liver, and homogenize the liver, and then you analyze the liver in order to test any toxins. When you boil or cook, some toxins can be detoxified, but there are toxins which will not be detoxified by cooking, especially BTOX, botulinum toxin, hard to detoxify by cooking. And also other, like, toxic elements, poisonous elements like arsenic, heavy metals, everything goes to the liver. An organ or part that belongs to both respiratory tract and the digestive tract. So the answer is pharynx, the first one. The buccal cavity is a part of digestive system. Even though we can use the buccal cavity to breathe in, it's not a part of respiratory tract, it's a part of digestive system. Larynx is a part of respiratory system. Trachea, respiratory system. So the answer is pharynx. Select the false statement. The cerebellum helps to maintain body balance. That is correct. Apart from the cerebellum actually coordinate all the voluntary movements. It's coordinate the voluntary movements. It doesn't control the voluntary movements, but it coordinates the voluntary movements. The voluntary movements are controlled by the cerebrum. Okay. Apart from the cerebellum, in our inner ear, we have semicircular canals, right? That also helps to maintain the body balance. That is why if you are being slapped hard, sometimes you will lose the balance. If someone slaps you, you may lose the balance because it affects the semicircular canals in your inner ear. So for the body maintain the body balance, cerebellum is important. That is correct. It's not a false statement, it's a correct statement. Reflex actions like lifting leg when step on a thorn is controlled by the medulla oblongata. That is wrong. Yes, there are reflex action controlled by medulla oblongata, but not these type of things. The reflex action like lifting the leg when step on a thorn is controlled by medulla oblongata? No. Like vomiting and all, yes, but not something like this. Not like these type of reflex action. They are controlled by what? Spinal cord. So this is the false statement. So this is the correct answer because this is the false statement. In a healthy person, all the glucose will be reabsorbed during the urine function. That is true. If you don't have diabetes, then all the glucose has to be reabsorbed. You can't waste. Bile is produced in the liver. That's also correct. It's produced in the liver, but temporarily stored in the gallbladder. Then through the bile duct, it released into the duodenum. Name a gas released due to eutrophication that directly contributes to the global warming. Methane gas. Methane gas. From the eutrophication, through the eutrophication, after sometimes these bacteria that causes the eutrophication also will start to release methane during the decomposition process. What is the product released due to the activity of nitrosomonas? that is nitrite. Nitrosomonas converts ammonium ion. This is biological nitrogen fixation. So nitrosomonas convert ammonium to nitrite. Then nitrobacter converts nitrite to nitrate. Biological nitrogen fixation. Okay, guys, the MCQs are over. We can move into the second paper, but do you have any questions from the MCQs? If not, we can quickly move into the essay type questions. Okay, then we will move into the essay type questions. Okay, Putala. The structured essay and the essay. Okay, now I heard Jagat also describe these things. So if you look at, you can say this pyramid X is what? It's the energy pyramid. Then you can see pyramid Z is what? This one. This is biomass. And this is the number pyramid. So name the ecological pyramids given as Y and Z. So pyramid Y is a number pyramid. If you want, you can just write the answers. And here biomass. Then what is a value suitable for blank A in the pyramid X? So you know that you have discussed already that 90% of the energy, when it moves from one tropic level to the other tropic level, 90% of the energy, will be used by the organism for, excretion to all the other activities. So only 10% goes to the next tropic level. So from 2000, only 10% goes to this. So it's 200 joules. Out of X, Y, and Z, which pyramid has or have the possibility to be inverted? Guys, remember, number pyramids can all easily be inverted. Why? Because if you look at the best example is, look at a mango tree or a coconut tree. Mango tree. Only one tree, but in the mango tree, there can be so many organisms, right? So, number pyramids, in the first tropic level, you may have only one organism. Then, there are other organisms. Or even if you look at the coconut tree, the same thing. There can be coconut beetles. Hundreds, thousands of coconut beetles can take food from the one coconut tree. So, one organism. Okay. So definitely number pyramids can be inverted. Apart from the number pyramids, even the biomass pyramids can be inverted rarely, not always, especially in the aquatic environment. You know why? See, now in aquatic environment, I will give you one of the best examples. A great white shark can eat lot of small fish. No, the lifespan of the small fish is very less when compared to the great white shark. Okay, so at a given moment, you may have less number of small fish. Even though you have about 100 small fish, it is not equal to the mass of the great white shark, right? Now, during the entire lifespan of the great white shark, it will eat thousands of small fish. But at a given moment, there are no 1,000 small fish. No. At a given moment, you may have 100 small fish and one great white shark. Or if you look at phytoplanktons, At a given moment, they may have only very small, less number of phytoplankton, the mass, because their lifespan is less. But the higher organism, their lifespan is more. So during their life, during their entire life, they will eat a lot of small fish. But then at that particular moment, there will be only 100 small fish and one large fish. So, you know, the top level may have a higher biomass than the lower level if you take one particular moment. In a particular given moment. Okay. So write a 4-linked food chain to represent the trophic level given above. So you can write anything because there is no picture or anything. Then you can think about, now since Jagasa had done that, I'm not going to explain about the food chain. So because he has explained it, no, otherwise we are taking more time. No, so you can just write any food chain. Maybe you can always, it should be start with the producer, primary consumer, secondary consumer, and the tertiary consumer. Each organism is known as a link. Producer, primary consumer, secondary consumer, tertiary consumer. What is meant by bioaccumulation? Actually, the word bioaccumulation is actually similar to biomagnification. So that means the accumulation of heavy metals and the toxins through the trophic level is known as bioaccumulation. Accumulation of heavy metals, toxins through the trophic levels is known as bioaccumulation. Then if you look at B, table gives some hypothetical data on the distribution of chronic kidney disease in one province in Sri Lanka. Represent this data graphically. So the best thing is, guys, for each year you can draw a bar graph, right? So if you look at 2011, it's only 250. So very small amount. But 2012, it's about 600, somewhere around here. Then it's 750, somewhere around here. So likewise, you can just draw a bar chart, okay, to represent the data. Then, according to the data given, what prediction can you make about the distribution of chronic kidney disease? So what prediction can you make? See what has happened. The incidence, the number of patients of the chronic kidney disease is increasing every year. So the prediction is that the number of kidney patients with chronic kidney disease will increase with the time. In 2016, there will be more patients. The number of the chronic kidney disease patients increase with the time. Then C, the diagram shows a sketch of the carbon cycle in the biosphere. Name the process X. Carbon dioxide is fixed by plants. So what is this process? Photosynthesis. Photosynthesis. Okay, guys. Now the second question, there is a problem. It's not the process X. It's actually process Y. What are the two types of process Y? What are the two types of process Y? Process Y is what? Now you can see this is carbon cycle. So you can see in the process Y, plants are releasing carbon dioxide. So plant releases carbon dioxide through respiration, right? So what are the two types of process X, process Y? Sorry, it's not X, it should be Y, should be corrected as Y. So there are two types, aerobic respiration and anaerobic respiration. Aerobic respiration and anaerobic respiration. Aerobic and anaerobic respiration. Aerobic respiration and anaerobic respiration. Two pollutants released by the combustion of fossil fuel. It's very easy. Sulfur dioxide can be released, carbon monoxide, oxides of, even sometimes nitrogen dioxide can be released, but sulfur dioxide is more prominent than the nitrogen dioxide when it's come to the especially coal. D, the diagram shows a setup arranged to show the carbon dioxide is produced in respiration. Which device should be connected to X for the function of this setup? So we have to connect a suction pump, or else you have to connect another beaker with a tap, to reduce the pressure, then it will remove it. So here the main best thing is making a suction pump. So even though a device mentioned in 1 is connected, this does not function due to a defect in it. What is the defect in it? In the sense, in this setup, what is the defect? See here is the defect. Always remember, the tube that goes out connected to the next one should never be put into the water, into the solution. Why? Because when you operate the suction pump, otherwise the water will be sucked out, not the air. So that is not the purpose of this, no. So this won't work. So you have to put this tube here, and then this tube you have to cut somewhere here. Okay, that is a problem in the setup. So after correcting this potassium hydroxide solution, guys, KOH solution is there to absorb carbon dioxide. You know that carbon dioxide is an acidic oxide, so that is absorbed by the potassium hydroxide. Colorless lime water in flask B. Now what happens? First, the air enters into this. So whatever the carbon dioxide in the normal air will be absorbed by the potassium hydroxide solution. Then it goes to the second one, B. In B, we have colorless lime water. So colorless lime water will remain colorless if no carbon dioxide is in the air. Okay. So to prove that there are no carbon dioxide in the air, we have to use the flask B, the lime water in the flask B. So once the air sucked in, air contains carbon dioxide, but that carbon dioxide will be dissolved here. So whatever the air goes out doesn't have carbon dioxide. So you can prove that using the colorless lime water in B. So to prove that there are no carbon dioxide in the air coming into the setup, to prove that there are no carbon dioxide, to prove that there are no carbon dioxide in the air entered into the setup. Entered into the setup. So then colorless lime water in flask D. Why do we need the colorless lime water in flask D? Now here, whatever the air comes into the setup doesn't have carbon dioxide. Okay, now whatever the air enters here doesn't have carbon dioxide. However, when the grasshoppers do the respiration, they release carbon dioxide. So then if the water, if the colorless lime water in D turns into the milk color, that proves the grasshoppers released carbon dioxide, because the air entered into the flask C didn't have any carbon dioxide, but whatever they went out from the flask C had carbon dioxide. So that is because grasshoppers release carbon dioxide during the respiration. So the purpose of having colorless lime water in D is to prove that carbon dioxide is present in the air coming from the flask C. Carbon dioxide is present. Carbon dioxide is present in the air coming from flask C due to the respiration of grasshoppers. If CO2 is present, it turns into the milk color or white color. Colorless lime water turns into the milk color or white color. Look at the second one. Now this is something related to the photosynthesis experiment. Parts of A, B and C. Which part stains blue-black when subjected to the iodine test for starch? Now, you can see here this is covered, but part A and C is not covered. So the covered part does not get the sunlight. However, if you look at the part C, look at part C. We have put that into a flask with potassium hydroxide. There is another issue with this, because potassium hydroxide absorbs carbon dioxide. So whatever the air inside this doesn't have any carbon dioxide. You have removed the carbon dioxide from the air because of the presence of potassium hydroxide. So this part of the leaf does not get carbon dioxide, no carbon dioxide. And this part doesn't get sunlight. This part gets both carbon dioxide and the sunlight. Okay. So when you subject this to the starch test, you would see that because there is no sunlight, no photosynthesis here. Because there are no carbon dioxide, no photosynthesis here as well. The photosynthesis will there in the part A. So it will give a reaction to the starch test. But in B and C, no photosynthesis due to lack of carbon dioxide in C and lack of sunlight to B. Guys, hope you understood. So what factor essential for photosynthesis was studied here? Carbon dioxide and sunlight both. Carbon dioxide and sunlight, CO2 and sunlight. Carbon dioxide and sunlight. State a reason for each of the following practices followed in the above experiment. Keeping the leaf for about 2 days in the dark. Why? Yes, to de-starch, to get rid of any starch, stored in the plant leaf. to remove or to get rid of, to remove any starch stored in the, because we know that what happens to the glucose after the photosynthesis. Now from the photosynthesis, the main product is glucose. No? So first, glucose turn into starch and temporarily stored in the plant leaf. Okay. After some time, this starch again converted into sucrose, because sucrose is more soluble in water. So it is easy for the phloem translocation, and also the plant cells will not take it for respiration easily because it's sucrose, not glucose. So because of that, glucose first turn into starch. Starch will be temporarily stored in the plant leaf, temporarily. But when you do this destarch, you can remove it. Then starch converts into sucrose and undergo phloem translocation. Okay. So now any test that we do to understand, or any test we do to check whether photosynthesis has occurred or not, what do? We test whether the leaf contains starch or not, because first of all, we take the plant, whether we are going to test carbon dioxide or sunlight, we have to keep it in the dark for 48 hours to get rid of any starch in the plant leaf. Okay, because always starch will be stored in the form of always starch will stored in the plant leaf. So we have to get rid of that first. So that is why we are keeping it for 48 hours in the dark. So once we get rid of the starch, then we do the test. We do the experiment for carbon dioxide or sunlight or whatever. So if photosynthesis happen, now here photosynthesis only happens in A, right? Photosynthesis only happens in A, only occurred in A. B doesn't occur because no sunlight. In C, it doesn't occur because there is no carbon dioxide. So A, the photosynthesis will occur. So you will get starch. Then you take this leaf, you will remove chlorophyll and you first boil it with water. Then you boil it in the ethanol using a water bath to get rid of chlorophyll. Then this looks like a white color one. Then you add iodine. So when you add iodine, if starch is present, it turns into blue-black color. If starch is not there, no color change. So here, A, since the photosynthesis has occurred because it got sunlight, carbon dioxide, everything, we have starch here. So it turns into blue-black color. But B and C, no color change with the iodine test. Then placing potassium hydroxide in the flask, what do we expect? To remove carbon dioxide. Now look at the next one. Hemophilic male married to a hemophilic female. Hemophilic male married to a hemophilic female. Guys, please change this. This question has a small issue. Just change this as hemophilic male married to a hemophilic carrier female. Healthy but carrier. Hemophilic carrier female. Okay. So please change it in your paper. If you have a hard copy, please change it. Otherwise, at least put a note. Otherwise, you are going to get confused. So hemophilic male and a carrier female. Puta, there are no hemophilic females. Hemophilic males are there. Only males are having the hemophilia. Okay. One person asked me the question. That is why I am answering it. So hemophilic male and hemophilic carrier female. Carrier female. Not a patient, just a carrier female. So Hemophilic male means now male means X and Y, no? Right? X and Y. However, now you can take hemophilic healthy gene as capital H. It's always located on the X chromosome. And hemophilic disease gene as simple H. So this hemophilic male means simple H and then Y here. X simple H and Y. Hemophilic male. Okay. Males only get hemophilia. Okay. The males only get hemophilia. I mean, females can have it, but it's very rare, and also they don't survive. They don't survive. So then carrier female means they have, anyway, female means 2 X chromosomes. Okay, 2 X chromosomes. One should contains capital H and the other one is simple H. Now females, because they have one capital H and one simple H, since they have a healthy gene also, she is a carrier female. They carry the disease because of the simple H, but they don't have the disease because they have a healthy gene. They are carrier because they have this mutated gene, but they are not hemophilic patient because they have a healthy gene as well, because the healthy gene is dominant over the mutated gene. Okay. Now it's very rare that you get something like this in hemophilia. It's very rare, and also they don't survive the fetal stage because they can't survive the childhood trauma. One thing is that. And also, even if in rare cases, if they live, they can't pass the, puberty, the females. So now these are the genotypes of the parent. So the gametes, now father can have X simple H, a gamete like this. Another gamete can have the Y chromosome. Only 2 types of gametes. No? Then the female also can have X chromosome with the healthy gene and X chromosome with the disease gene. Okay. So these are the gametes for the female. So what happens? From father, the sperm can have X chromosomes with mutated gene. That can go with an egg having X chromosome with a healthy gene. So the result is X capital H, X simple H. From father, X simple H, from mother, capital H. So this is healthy daughter, okay. XX means daughter, no, sorry, healthy daughter but a carrier. Okay, healthy but carrier daughter, healthy but carrier daughter. You can say actually carrier daughter, hemophilic carrier daughter, hemophilic carrier daughter. They don't show the disease because they have a capital H, but then they are carriers because of the simple H. Okay. Then again, another sperm, give me a minute. Sorry. Y. So this X also can go with this one, no? This gamete. No? So in that case, X simple H, X simple H. So usually this fetus will not survive a disease daughter. Okay. Then hemophilic daughter. Hemophilic daughter. Then Y can go with this. X, this is healthy from mother. Then from father it's Y. So this is healthy male. Carrier female. Hemophilic female. Healthy male. Then again, here, because this X has simple H, here you have Y. So here, hemophilic disease male or hemophilic male. Hemophilic male. 1:1:1:1. Carrier female, hemophilic female, healthy male, hemophilic male. All four possibilities are there. 1:1:1:1. Guys, understood? Okay. Write 3 uses of gene technology. Now this is very easy to to make recombinant food, to make recombinant like in DNA fingerprinting, to identify criminals, to produce medicines, vaccines. We can use gene technology. Okay, there are so many uses of gene technology. C, name a double membrane organelle which cannot be seen in animal cell with its function. Very easy, double membrane organelle, but cannot be seen in animal. Now there are, nucleus has a 2 membrane, mitochondria has 2 membranes, chloroplast has 2 membranes. These are the 3 double membrane organelles. Okay. But they are asking name a double membrane organelle which cannot be seen in animal cell. But now if you look at nucleus and mitochondria can be seen in both animal as well as plant cells, right? Nucleus and the mitochondria. So only double membrane organelle which cannot be seen in animal cell would be the chloroplast. Okay, chloroplast. So the function is what? Photosynthesis. Function is photosynthesis. Now, if you look at here, given below is a diagram of jasmine, a plant treated to obtain another plant from it easily. What is the name of the above method propagating plants? This is the ground layering. What do you do when you do the ground layering? You have to put a cut here in the cambium, in order for this branch to develop the roots. Okay, that is how you do it. So this is ground layering. Ground layering. Ground layering. Okay, I hope you guys are writing the answers. Name a method that can be used to produce large number of identical plants from one source. Plant tissue culture. Plant tissue culture. You can get thousands of identical plants from one particular source if you use the plant tissue culture. So the second one answer is plant tissue culture. Third one, look at the diagram of 3 kinds of fruits and seeds that are dispersed by different methods are given below. You can easily say, you can see the wing-like structure here, right? So this is from wind. And here you have the fruit. the succulent part and everything. So by animals, dispersal methods are this contained fibrous pericarp. So from water, this can float in water. So wind, animal and water. These are the fruit and seed dispersal methods in these ones. Given below is an illustration of the menstrual cycle occurring in the reproductive system of a sexually mature female. Now, if you look at here, Putala, if you look at here, guys, this gives you the follicular phase and the luteal phase. The follicular phase and the now menstrual cycle occurs in two locations now, right? What are the two locations where the menstrual cycle occurs? Ovary. In ovaries it occurs, and in the uterus it occurs, right? Ovary and the uterus. Here, this is for the ovaries. The follicular phase and the luteal phase is for the ovaries. In the uterus, what are the phases? Menstrual phase. Guys, give me 1 minute. 1 minute. Sorry. So, if you look at the uterus, there are three phases: menstrual phase, proliferation phase, and the secretory phase. Menstrual phase, proliferation phase, and the secretory phase. Okay, now this represents the ovary. Okay, here we have follicular phase and the luteal phase. Now, in the above figure, which phase in uterus comes next to C if no fertilization occurs? Now, here during the follicular phase, the follicles will develop, and then at B, ovulation occurs. Then during here, what should happen is the fertilization should occur during the luteal phase from B to C. Actually, it's not entirely moved till the C. Fertilization should occur like, after 48 hours after ovulation. So it occurs in the, usually fertilization occurs in the fallopian tube. So now in order to have a successful fertilization, sperms should be released into the female reproductive system at least 48 hours before the ovulation or after the ovulation. That may lead to the during this period. But if it is more more than 48 hours it took, then successful fertilization may not occur. Okay, so then, however, now if the fertilization doesn't occur, what happens in the uterus? Now they are asking in the above figure which phase in the uterus comes next to C if no fertilization occurs. They are not asking about the ovary, they are asking about uterus. So what will happen if fertilization doesn't occur after C? Menstrual cycle will restart. So after C in the uterus, the menstrual phase will start. Because at the end of the C, again menstrual cycle has to restart. So in the uterus, after C, the menstrual phase will take over. Okay, menstrual cycle or menstrual phase will take over about 4 days, the menstrual period. Pudala understood? Any questions from this? Any part you didn't get? Okay. According to the above figure, during the period between which two letters can a fertilization occur? B and C. One person asking whether we can about the phase, can we write the uterine wall degenerate? No, you have to write the proper terminology. You have to use the proper terminology. Yes, during the menstrual phase, the uterine wall, it breaks and moves out. No, but you have to use the correct terminology. So that phase is known as menstrual phase. So you have to write the proper words. Okay. Yes, we know that during the menstrual period, the uterine wall that breaks and moves out, but the correct wording is menstrual phase. So in the above figure, which phase in uterus comes next to C is the menstrual phase. Then according to the above figure, during the period between which two letters can the fertilization occur? Between B and C. What special event occurs in the ovary in the occasion of B? Ovulation. You know what happens is there will be a high concentration of luteinizing hormone. Due to the high concentration of luteinizing hormone, the ovulation will occur. The ovulation is regulated by luteinizing hormone. That means LH hormone secreted by the pituitary. So the concentration of LH hormone suddenly rises when closer to the ovulation. So at this point, see, this shows you the LH hormone level. This is LH hormone, this is FSH hormone. So due to the sudden increment in the LH hormone, the ovulation will occur. Ovulation in the sense, releasing an ovule to the fallopian tube. So what special event occurs in the ovary in the occasion B? Ovulation. What are two hormones involved in the two phases given in the diagram? So FSH hormone and the LH hormone. What is the secretory organ of these hormones? Pituitary gland. Pituitary gland. The hormones are FSH and LH. FSH and LH. Follicular stimulating hormone and luteinizing hormone. Then which secrete that? Pituitary gland. Pituitary gland. So state an importance of this type of division to keep the constant number of chromosomes through the generations. So this to keep the constant number of chromosomes. To keep the constant number of chromosomes through generations. To keep the constant number of chromosomes through generations. Identify the problem in the above diagram. If this is meiosis, actually it should result in 4 daughter cells. So that is incomplete. This diagram is incomplete. There should be 4 daughter cells. There should be 4 daughter cells. The blood pumped from the human heart circulates in 2 routes, systemic and pulmonary circulation. State 2 differences between systemic circulation and pulmonary circulation. Guys, if you look at, now if we draw the heart like this, so we know that our heart has 4 chambers, right? So left atrium, left ventricle. Usually right hand side we put the left, Left hand side, we put the right part, no? Right atrium and right ventricle, isn't it? So right atrium, there are two blood vessels, superior vena cava and the inferior vena cava, which brings the deoxygenated blood to the right atrium, right? It brings the deoxygenated blood to the right atrium from the superior vena cava, upper part of the body, inferior vena cava, lower part of the body. And in between right atrium and right ventricle, we have tricuspid valve. Tricuspid valve. How do you memorize it? Right, right, right. In Sinhala, we call it dakuna. Right? Right means dakuna. So, 3 letters. Dakuna, 3 letters. So, tricuspid. Tricuspid valve. Tricuspid valve. So, from the during the atrial contraction, this deoxygenated blood came from the superior vena cava and the inferior vena cava, released into the right ventricle. Then right ventricle is attached to pulmonary artery, which takes the deoxygenated blood to the lungs. Then in the lungs, the blood will be oxygenated, and oxygenated blood will come to the left atrium through pulmonary veins, left pulmonary vein and the right pulmonary vein. So the oxygenated blood will be filled in the left atrium. Then during the atrial contraction, again, it goes to the left ventricle through the bicuspid valve. Bicuspid. Left means vama. It has 2 letters. So bicuspid. Bi means 2, no? Bicuspid. Then left ventricle, it will be filled. Then during the ventricular contraction, it enters into the aorta and distributed throughout the body with a very high pressure. So you can see we have 4 chambers: right atrium, right ventricle, left ventricle. So deoxygenated blood from the superior vena cava and the inferior vena cava comes to the right atrium. During the atrial contraction, it passed to the right ventricle. Okay. Then during the ventricle contraction, it enters into the pulmonary artery and goes to the lungs. Still the deoxygenated blood. Then in the lungs, it will be oxygenated. Then through the pulmonary veins, oxygenated blood comes to the left atrium. Now left atrium has oxygenated blood. Then during the ventricular contraction, it enters into the left ventricle. Then during the left ventricle contraction, during the ventricular contraction, from the left ventricle, it enters into the aorta to distribute throughout the body. So systemic circulation is this, pulmonary circulation is this, which happens between the heart and the lungs. Heart and the entire body of entire other organs is systemic circulation. So the differences would be, first thing is, in the systemic circulation, it distributes blood throughout the entire body, but the pulmonary circulation distribute or takes blood only to the lungs, or it will distribute the blood between the heart and the lungs. But the systemic circulation throughout the entire body from the heart. Okay. Second thing is you can talk about the pressure. Systemic circulation has a high pressure, or the pressure inside during the systemic circulation is higher than the pulmonary circulation. In the systemic circulation, oxygenated blood will move out from the heart. In the systemic circulation, oxygenated blood moves out from the heart. But in pulmonary circulation, deoxygenated blood pumps out from the heart. So systemic circulation, heart pumps out oxygenated blood. oxygenated, while pulmonary circulation, heart pumps out deoxygenated blood. So these are the differences. Sometimes a blood clot may block a blood vessel in the brain, leading to destruction of the nerve cells. State a symptom of this. There are so many. Paralysis. You can, entire part of your body or entire body can be paralyzed. You can feel the dizziness, vomitation, and everything. First, those are the small symptoms, but the main problem is the paralysis. But if it occurs, if you don't treat it properly, not only paralysis, even it can lead to the brain death. It can lead to a coma. Guys, 1 minute. Give me 1 minute. Sorry. So calcium level in the blood of a person was reduced in a considerable amount. Excessive secretion of which hormone can cause this? It's actually secreted by thyroid gland. It's a calcitonin. Calcitonin reduces the calcium level in blood. So if you have excess amount of calcitonin, the blood calcium level will go down because it will deposit all the calciums in the bones. Answer is calcitonin. Calcitonin. What is the functional unit of nervous system? Now, this is a very important thing. Usually we say cell is the functional and the structural unit of tissues. No? But in the nervous system or nervous tissue or nervous system, the functional unit is reflex arc. Yes, reflex arc. Some people gave correct answer. You know reflex arc, right? Guys, don't get confused with the reflex arc and the reflex action. Those are two different. Reflex action also follows the reflex arc, but not only reflex action, but any nerve, any response to any particular stimulus follows the reflex arc. So reflex arc means from the sensory organ, it is detected by the sensory neurons. Then sensory neurons brings it to the central nervous system, where you have the intermediate neurons. Then from the intermediate neurons, it goes to the motor neuron. Okay, like this. Now you have the sensory organ, maybe eye or something like that. So then it taken by the sensory neurons. Sensory neurons. Then you have intermediate neurons in the central nervous system, intermediate. Intermediate. Then you have motor neuron. Goes to the effector. It can be a muscle or a gland. Usually effectors are either muscle or a gland. So this is the reflex arc. Even in reflex action or any anything to do with the nervous system. It can be a reflex action. It can be a normal reaction to a normal stimulus. Everything follows the reflex arc. So don't get confused with the reflex arc and the reflex action. Name the structures which act and be out of X, Y, Z. Answer the following questions by using the letters given in the above diagram. So as B out of X, Y, Z, which act as the B? Z, right? Z neuron, intermediate neuron. So B is what? Intermediate neuron. The neuron which transmit impulses to the central nervous system from the receptor is Y, sensory neurons. This is a sensory neuron. So this is Y, this is Z, this is X. Okay. So in the reflex arc. So heart, in the heart muscles, the cardiac muscles, guys, it's not unnucleated, it is uninucleated. Okay. So yes, heart muscles are uninucleated, but in stomach we have smooth muscles. So they also uninucleated. In heart also we have uninucleated cells. In stomach also we have, because stomach contains smooth muscles. Heart contains cardiac muscles. Striations, yes, in heart cardiac muscles, but in stomach we have smooth muscles, they are not striations. Voluntary, no. Voluntary, no. Branched, yes, but smooth muscles unbranched. So if you look at heart, we have cardiac muscles, they are uninucleated, striated, but non-voluntary. However, they are branched. In stomach, we have smooth muscles, again uninucleated, non-striated, not striated, non-voluntary, it's not branched. Which of the above organ has a muscles which never fatigue? Heart. That is cardiac muscles. Can't get fatigued, right? Name another organ which has a type of muscle tissue found in the stomach. So the smooth muscles, where can you find smooth muscles? In the blood vessels, in the bladder, urinary bladder. Okay. Many organs, even our esophagus, the smooth muscles. So here you can see when the normal glucose level goes up, hormone X is secreted, then it comes to the normal glucose level. So once the glucose level goes up, hormone X is secreted. That is what? Insulin. Insulin. Once insulin is secreted, it will come back to the normal. When the normal glucose level goes down, Y is secreted, which is glucagon. So it will again bring back it to the normal level. So insulin reduces the glucose level, while glucagon will increase the glucose level. So X is insulin, Y is glucagon. In which part of the digestive system the digestion of carbohydrates starts? So if you look at the digestion, Putala, digestion, two types of physical digestion and the chemical digestion. Anyway, the physical digestion starts in the buccal cavity. And also the chemical digestion of carbohydrate occurs in the buccal cavity. In the buccal cavity, we secrete salivary amylase, right? Also known as ptyalin, salivary amylase. Salivary amylase will convert starch into its constituent sugars like maltose, starch, starch to maltose. So it starts in the buccal cavity. It will not be completed, but it will start. Write down adaptation in small intestine wall to increase the absorption of nutrients. So you can see it has a brush border, right? And it's folded into villi and the microvilli. And microvilli forms the brush border. So it increases the surface area to absorb the villi and the microvilli in the villi and microvilli in the uterine wall increases the surface area. The second one is there is a blood supply, proper blood supply in order to absorb the nutrients. One thing is it has increased the surface area by villi and the microvilli, and then there is a blood supply in order to absorb the nutrient. In which part of the digestive system the lipid metabolism starts? Lipid metabolism starts, especially starts in the duodenum, right? In the duodenum, the bile pigment secreted into the food, and it causes the emulsification of lipid. That means lipid will be broken into tiny droplets in order to increase the surface area. We call it emulsification. That occurs in the duodenum. State whether secretion of ADH increases or decreases in the following instances. So what is ADH? Where exactly ADH is secreted? ADH stands for what? ADH stands for what? ADH, you remember, right? So ADH means anti-diuresis hormone, anti-diuretic hormone, ADH. It's also known as vasopressin, and it's secreted by actually our pituitary. Actually, it's released by the pituitary, but the produced in the hypothalamus. It's released by the pituitary, but it's actually produced in the hypothalamus. So this ADH, what does it do? ADH is important in order to maintain the water levels of the body. So if we have high, like, if the what do you call, if our in our body, if we have less water in our body fluids, if the concentration of solute concentration, salt concentration increases, what happens is ADH will be secreted, like if you look at now ADH hormone. Now imagine in your blood, the blood volume drops, reduces. That means not enough water. So the concentration, salt concentration and everything will increase. So that will send the signals to the brain. So the hypothalamus will produce ADH hormone. It temporarily stored in the pituitary, and then pituitary secretes the ADH hormone. Then what happens? ADH hormone will increase the reabsorption of water. So now the question is, state whether the secretion of ADH increases or decreases in following instances. When the water level of blood goes up, that means you have enough water, no? So you don't need to reabsorb. I mean, you can reduce the reabsorption. So to reduce the reabsorption, ADH level decreases. However, when the water levels goes down in the blood, then ADH level will increase so that the reabsorption will increase. So the reabsorption will increase. When the water level goes up, ADH level decreases so that the reabsorption will decrease, reabsorption of water. When the water levels in the blood goes down, secretion of ADH will increase so that it will increases the reabsorption of water. ADH increases means it increases the reabsorption of water in the kidney. When the water levels goes down, which process in the nephron helps in the regulation? Selective reabsorption. Selective reabsorption. You have to write it properly. Selective reabsorption. Selective reabsorption. Which part of the brain involves in the regulation of the body temperature? Hypothalamus. Hypothalamus regulates the body temperature. Then what is meant by reflex action? What is meant by reflex action? In the reflex action is what? It is a way that the body automatically and rapidly. It is a process in which the body automatically and rapidly respond to a stimulus. It is a process in which body automatically and rapidly responds to a stimulus. Rapid response to a stimulus. This rapid response is required to minimize any damage. Okay, it's a rapid response to a stimulus. Reflex action. And the last question, what is meant by, then the mentioned two examples of cranial reflexes. Cranial reflexes occur in the medulla oblongata. That means the brain stem. Okay, so there are two different types of reflex action, cranial and the spinal. Cranial examples would be sneezing, blinking of eyelids. Then the spinal reflexes would be, if you touch a hot surface, you take your hand off. If you, step on a thorn, you take your leg. Understood? Okay, Bhutala. So I think we have discussed the paper. Any questions? Any questions? So you had asked several questions. I answered most of them. So, yeah. Okay, then I think Jagat sir can take over. Jagat sir. Guys, give me a minute. If you have any questions now, you can post while till Jagat sir starts. I can answer the questions if you need any. If you have any specific questions, I can quickly answer 2 or 3.
Right. I think if you have any questions, you can ask from Chanda sir before he leave. And we have nearly 1 hour more left, and I will continue the remaining questions of the other paper. You have any questions, or you can raise the hand, or you can put into the chat if you have any questions to ask from Channa Taranga.
Okay, if there are no questions, then fine, because I got several questions, so I answered them during the session itself. So then, okay.
Okay, sir. Right. Okay, sir.
Yes, sir.
Very much
Okay.
for sharing your valuable time today.
Okay, thanks.
Yeah.
Okay,
And good night.
good
And
night.
God bless you. Yeah.
God bless.
Right. Okay, students,
Okay.
you can. We will continue the remaining 1 hour. So with the other some important questions I discussed in the other paper. I'll share the screen now. So I discussed the first question completely. This question I discussed completely, and second question. Question number 2. Identify each biomolecule that build the living body, which described by the following statement. So formed by fatty acid and glycerol, that is lipid. So there are 4 biomolecules: carbohydrate, protein, lipids, and nucleic acid. To A, the answer is lipids. And the structural unit of amino acid, so that is the biomolecule, will be protein. In other words, amino acids are the building blocks of protein. C contain the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. So there are 5 elements present in that is called nucleic acid. Nucleic acid. Next one, the most abundant organic compound in the living matter, that is carbohydrate. That is part A. And part B. The cell is the smallest structural unit of the living body, and it is a functional unit of the living body as well. Identify the cell types of B. So B actually plant cell. So this is given in your grade 10 textbook chapter 6. B represent plant cells. The difference between the cell A and B. So A cell wall is A actually animal cell. A represent animal cell. So if you, one of the best example, cheek cells. Under your syllabus, there's a practical observing cheek cells and observing onion peel. The plant cells. So A animal cell, B plant cell. Therefore, you can write A cell wall. One difference, you can write cell wall present in B but absent in A. Cell wall is present in B but absent in A. Second difference, this is one difference. The other difference, you can write large vacuole present in B. Large vacuole present in B but absent in A. Absent or not present in A. So those are the two main differences you can see here, cell wall and the vacuole. Part 3, write 3 steps. How do you prepare a side of an onion peel cells? So this is a practical. Remember, your structure is a even compulsory questions. Your 25% you are getting from the practicals. So how to observe onion peel under the microscope? This is given in your grade 10 textbook chapter 6. Observing onion peel, and there's a video also about that. Wait, I'll show that video, then you can easily remember that. Give me a second. I'll share the video. Observing. Okay, you can see it now.
The two pieces apart. There's a thin, transparent layer of cells holding these two pieces of the onion layer together. That's the layer of epidermal cells that I'm looking for my specimen. Now I need to obtain a piece of that onion epidermis to place on my slide. I can do that by tearing it off with a pair of tweezers. This is a very delicate, thin layer of cells, and they'll fold over on themselves. So when you place that onto the slide, once you get it off your tweezers, you're going to need to spread it out a little bit. The little piece of onion epidermis can easily get all wadded up, and that makes it very difficult to see the cells. While filming in the lab, I forgot to include, and so I'm going to show it to you here. The best way to get a large, flat sheet of these onion epidermal cells is to peel that layer of onion skin off directly onto the slide. It's very thin, it'll wrap right around the slide just like saran wrap. So apparently I've lost. Head back to the lab. For this wet mount, I'm just going to use iodine. This will provide the wet part of the wet mount, and it will also stain the cells and make them easier to see. Now I'm going to obtain a cover slip, a clear square, that I'm going to place on top of my specimen on the slide. The best way to apply that cover slip is to hold it at a right angle, let it drop, let it fall. As it falls, it will push the air out and you'll have fewer air bubbles this way. Here's my finished, prepared specimen. Here's a closer look at the application of that cover slip. Again, I hold it at a right angle to the slide, I let it drop, That pushes the air out. Sometimes I'll give it a little tap, and now I'm all set. I'm now going to examine my specimen using the 4x objective lens that will magnify the specimen 40 times actual size. Here's my first look microscopically at the onion epidermal cells magnified 40 times. These cells look kind of like misshapen bricks, a whole bunch of them. The compound microscope is parfocal. What that means is once I get my specimen in focus at one power, in this case the scanning power, I can switch to the next higher objective, the low power in this case, and my specimen will still pretty much be in focus. I'll only have to adjust the focus a little bit to get it crisp. Here are my onion epidermal cells magnified 100 times actual size. So this is a closer look at that application. To make it more view, adjusted the light level. I turned it. The first way is later or through your nose like a banded lens. Here are a couple examples of actual size. These samples, I can see the thick cell walls and these elongated cells.
Okay, you can see the screen now. How to these practicals are very important. You have to pay more attention to these practicals when you are studying the textbook. Especially each lesson there's a activity 1.1 no 1.2 both 10 and 11 textbook, because you will be getting nearly 25% in the structured essay from these practicals, the activity based questions, and sometimes you might have not done this in the school yet. Therefore you have to go through at least you have to go through the materials, method, observations, conclusion, and sometimes they can give the steps in the wrong order. They'll ask you to arrange in the correct order. Sometimes they can give the steps with with one step missing, or maybe two vital steps missing. They'll ask what is missing in that procedure, or they'll give the diagram with defects and ask what is the error in the setup and how to correct it, or any defects in the given procedure. So that's how you can get the questions. Otherwise, they will never ask you to write the entire experiment from the beginning. Right. So you can see the onion peel. This is observing cheek cells under the light microscope, and the other. This is the question is about the observing onion peel. So you can cut onion and obtain inner fleshy tissue. So they ask in the question. If you move back to the question, they ask here. Write down in 3 steps how to prepare the slide of the onion peel. So you have to write that in 3 steps. So this is what you should include. Remove a peel from the inner surface or outer surface and transfer to a watch glass, and then transfer that to a slide. So first you can write this. Look at the screen. You can see now. Remove a peel from the inner or the outer surface of and to contain water. That is the first step. Number 1. Number 2, you can write put water drop to a clean glass slide. Put a water drop. Number 2. Number 2, the pink one. Number 2. Number 3, This is number 2. And last step you can write cover it with the cover slip and without rubbing air and bubbles and observe it. So these are the 3 steps. I'll highlight the last one. This is the last step. Third step. Step 3. Number 1, remove onion peel from the inner outer surface it and transfer it. Why do we immediately after taking out we have to put into water? To prevent the cells getting dried. To prevent the cells getting dried. And you cover that with the cover slip again to prevent it's getting dried. Otherwise you will not get a clear look, or you will not be able to observe it clearly. And without tapping the air bubbles, very important. So these are the 3 steps. Right. Then next one, let's move back to the again. So this one done. Is done. Part 4a, identify the following methods of cell division. This is very important. Please put 3 stars for this. This is introduced in new syllabus in 2016. Most frequently they ask questions from this part. This diagram also very important about methods of cell division. So this method of cell division is known as meiosis. Why? 2n become n, number of chromosomes become half. So this method of cell division called meiosis. What is indicated by X and Y? X will be division 1, that is meiosis actually meiosis, and Y will be mitosis. It begins with meiosis and it's ended up with mitosis. Here you can see 2n to n, that's why it's meiosis. X. And next one you can see the number of chromosomes no change, n to n, say 2n3 to 2n3 like that. That is why it's mitosis. So Y will be mitosis. Or there are 2 steps or 2 stages here. Mitosis. Next one, write an instance where the above method of cell division occurs. So this type of cell division occurs during the formation of gametes. Formation of gametes or sex cells. Formation of gamete cells. In the formation of gamete cells. This happens in the formation of gamete cells. Right. Next one, part C. This is also very important. So far in oral past papers they haven't asked this practical. This is also grade 10 experiment. I told you this time oral paper you will be getting more from the grade 10 practicals. Grade 10 work. Structured essay you will be getting more from the grade 10 work. Structured essay you will be getting more from the grade 10 work this time. I'll erase the screen. Part C. The following setup kept for a few days. What are the purpose of including KOH in this setup? So KOH is a base. It can absorb what gases? Acidic gases like CO2. So CO2 absorbed by KOH. Therefore, to absorb CO2 inside the flask. To absorb or else to remove. To absorb CO2 in the flask. To absorb CO2 inside the flask. Or else you can write to remove CO2 inside the flask. An observation in this setup. Colored water level rises up in setup apparatus A but not in B. You can write here what is the observation here. Colored water level rises up in setup A. In A, in the glass tube, but not in B. Why? Because B, no KOH. What happens here? CO2 germinating seeds release CO2 and absorb O2. Therefore, no change of the volume of air inside. CO2 given out, equal amount of O2 is absorbed. Therefore, no net change of the volume of gas inside the apparatus B. But here what happened? Oxygen is absorbed, but CO2 cannot fill that gap because CO2 also absorbed by KOH. So there CO2 is absorbed by KOH. CO2 released by the germinating seeds, absorbed by actually in the KOH also actually it absorbs CO2 released by the germinating seeds. We assume one assumption here: initial CO2 in the flask neglected. At the beginning also this flask is filled with air, atmospheric air contains CO2, but that CO2 we ignore here. Therefore, what happens in flask A? CO2 released by the germinating seeds, absorbed by KOH. Therefore, the reduction of the volume in the apparatus A is due to the utilization of oxygen. So there respiration occurs in the apparatus B also, but it cancels off because of CO2 given out when O2 is absorbed. So actually what happened? What is the aim of this experiment? To show that oxygen is absorbed during respiration. Colored water level rises up in the tube of A but not in B. Next one, name an assumption made here. One assumption is what? O2 absorbed is equal, volume of O2 absorbed for respiration is equal volume of CO2 given out. That is one assumption. Volume of O2 absorbed equal volume of CO2 given out. This assumption definitely they'll ask if you get a question from this O-level paper. If they give this question, definitely they'll ask the assumption. I'll highlight the assumption here. It's given in your textbook also. This is from which lesson? Grade 10, chapter 8. If you only write here, this is from grade 10, unit 8. Volume of CO2 absorbed equal volume of CO2 given out. That means it's balance. Release. And the other one you can write, the other observation here, assumption, initial amount of CO2 is negligible. Initial amount of CO2 in the flask is negligible. Initially also there's a little amount of CO2 in the flask, that's negligible or neglected. Part 4, what is the living characteristic that explained from this conclusion? What are the living features? Or is respiration, right? Part 4, the answer is respiration. Which living characteristic is represented here? Respiration. Instead of germinating seeds, you can have even grasshoppers, but germinating seeds actually it respire. You can't put boiled seeds here. If you put boiled seeds, no respiration. So living things. What is the observation here? Colored water level rises up in this but not in this. So this practical is very important, introduced to new syllabus. So far they haven't asked this in the past papers. Right, then I erase this. Question number 3. Modern classification was introduced by Carl Woese. What are the 3 domains? Three domains, very important. I think Chandas also explained about this. Domain archaea, domain bacteria, and domain eukarya. Domain archaea, bacteria, and eukarya. 3 domain. Next one. What are the kingdoms to which domain that eukaryotic organism belong can be divided? So kingdoms. So eukarya, there are kingdoms: kingdom Plantae, kingdom Animalia. There are mainly 4. I'll write them. Kingdom Plantae, that is eukarya. Organized nucleus present. Kingdom Animalia. Number 3, kingdom fungi. Next one, kingdom Protista. Kingdom Protista. This kingdom Protista consists of what? Algae and protozoa. Algae plus protozoa. Kingdom Plantae, kingdom Animalia, kingdom Fungi, and kingdom Protista. Number 3, to which domain living organisms in extreme environment belongs? That is domain archaea. Domain archaea. Write 2 features of fungi. Fungus, heterotrophic. Cell wall is made of chitin. Right here, fungi, cell wall is made of chitin. That is one important feature. Second one you can write, they are heterotrophic nutrition. Heterotrophic. No chlorophyll. Non-photosynthetic. Write 2 uses of fungi. There are many uses of fungi. You can write production of antibiotics, act as decomposers, used in bakery industry. Example, yeast. In addition to that, even some fungus used as food for man, agaricus, mushrooms. Some fungus used to make marmite. Production of ethanol. We use fungi. Right, that is 3A. Next one, 3B. The most important plant of the plant sexual reproduction is the flower. Name the parts of the given flower. So your chapter 14, grade 10, chapter 14 reproduction lesson, reproduction in man and reproduction in plant. Plant reproduction, sexual and asexual. Asexual also there are 2 types, natural asexual method and artificial or man-made one. Artificial asexual reproduction, grafting, layering, tissue culture and stem cutting. Natural methods, there are 9 methods. Natural asexual method in plants: by roots, by bulbil, by underground stem, by rhizome, by comb, by leaves, by rhizome. There are various methods: by aerial buds, by runners, by suckers. Natural methods. So therefore, you have to go through that part also important. You can definitely get a question either essay or essay type or structured essay, if not at least few MCQs from the reproduction lesson, plant reproduction. So you have to name the parts here. So number 3, I'll put it was XYZ. X act as stigma. Y will be the style. Z will be the ovary. This will be the pedicle. Number 2, sepals. Collection of all the sepals sometimes called calyx. Number 4, filament. Number 5, anther. So both these together known as stamen. Both anther and filament together known as stamen, or the male part, or the androsium. What is the other term is for this? Androsium. Number 3, petal. But all the petals together known as calyx. But in the exam they will not ask all these parts. Some of these parts they can ask. So is this unisexual or bisexual? It is a bisexual flower. Why it is bisexual? It consists of both androsium and gynosium. It consists of both androsium and gynosium. They are asking give reasons. Consist of both, write the reason. Consist of both male and the female part, or else consist of both androsium and gynosium. Consists of both androsium and gynosium. Next one, what is the special name of the part XYZ together? XYZ together called, so you can see here this is X, that is stigma, Y is style, Z is ovary. So all three together known as pistil or carpel, or sometimes we call it gynosium. Or sometimes we call it pistil. Sometimes we call it carpel. Any of these accepted? Next one, the process of depositing matured pollens on the flower is known as stigma pollination. Right? What happens to the ovary and the ovules at the end of after the fertilization? So they undergo fertilization. What happens to the ovary and the ovules at the end after pollination? So then actually they are seeing the ultimate result. Ovary become the fruit and ovules become the seeds. After pollination, ovary and ovule, so that means ovules get fertilized. After fertilization they become the fruits and seeds. Number 5. Write 2 adaptations that can be seen in the flowers to avoid self-pollination. So one of the best example we can write presence of unisexual flowers. To avoid self-pollination, having unisexual flowers. Having unisexual flowers. Unisexual flowers. That is one adaptation. Bisexual flowers promote self-pollination, to avoid self-pollination. That is number 1. The other one you can write, number 2, second one, self-sterility. Self-sterility. So next one, parthenogenesis. So what is the process known as? What process is known as? That means occurrence of seedless fruits. That means transforming the ovary into a fruit without the fertilization of seeds. Transforming ovary into a fruit without fertilization of ovules. Without fertilization of ovules. If ovules are fertilized, you get seeds, but here without the fertilization of ovules, that means transforming of the ovary of a flower into a fruit without the fertilization of ovules. That is called parthenogenesis. Example, this happens naturally in pineapple, banana, breadfruit. Occurrence of basically occurrence of seedless fruits. What is that called? Occurrence of seedless fruits. Next one, you asked to write the method of the dispersal of Nagadharana by animals. Hora by wind. Seamango by water. Kudalu by explosive mechanism. I write here Nagadharana by animals because it has hook seeds. Hora by wind. Seamango by water. Kadu. Kudalu by explosive mechanism. By explosive mechanism. By the plant itself. Someday you can get this under MCQs also. Hora by wind. Seamango by water. And Kudalu by explosive mechanism. Name 4 factors that are essential for seed germination. Seed germination, number 1, water is necessary. Number 2, air or oxygen is necessary. Soil air. Air, or you can write oxygen. Soil water, soil air. Number 3, viability of the seed. Viability of seed. Or else seed must be matured enough. Matured embryo. In other words, we call it matured embryo. Number 4, temperature. Suitable temperature. If you put the seeds in the refrigerator, freezer, it will not germinate. Temperature, oxygen, air, water, or you can write H2O, or you can, instead of water, you can write moisture. So these are the 4 factors. If even if all these factors are available, if seeds do not germinate, even though all the necessary factors are fulfilled, some seeds do not germinate. We call it seed dormancy, bija suptatavya. Some seeds fail to germinate even though all the factors are fulfilled. What is that condition called? Seed dormancy. Seed dormancy. So these are the factors essential. Sometimes if you want to write here, some plant hormones also essential, right? Like gibberellins, you don't remember the name of the hormone, but sometimes the plant hormones also required for this. Done. Universal copy done. Shall I read this? Right, next one. Meaning your copy down. Okay. If you want, you can take even a screenshot. When you're done, please lower your hand. Done, right? The next one. Question number 4. I told you this photosynthesis experiment very important for the structured essay question number 2. Structured essay question number 2, most of the oral past papers they have asked photosynthesis experiment. There are 5 experiments. Testing whether carbon dioxide necessary, testing whether water necessary, testing whether sunlight necessary, testing whether chlorophyll necessary for photosynthesis. For 4 factors there are 4 experiments, but only for one factor we are not doing that in the school laboratory, that is water. Necessity of water cannot be showed in the school lab. Other three factors we can demonstrate, or we can show that it's necessary for photosynthesis, water, except water, carbon dioxide, sunlight and chlorophyll. Other than that, there are two other experiments: the starch test, testing the leaf for starch, and the other one, oxygen given out during photosynthesis, that we use a tube, the funnel tube experiment, that is aquatic plant, oxygen bubbles are evolving underwater. So therefore one of this experiment, this time this chlorophyll one bit important, and oxygen liberating one also bit important. Right. First one, what is the variable factor used in this test? Carbon dioxide. Sorry, sunlight. Sunlight is a variable factor. So the control test out of this, that is B. Control test is B. Number 3, it's not hard, right? So variable, what is variable for this? Sunlight. Sunlight is available for B, but not for part of the leaf A. So the control experiment will be B, and the experimental setup will be A. Next one, what is the purpose of keeping the plant in the dark for 48 hours? To de-starch leaves. Before most of the photosynthetic experiment, the starch already produced, we have to remove. Otherwise, it's like this: immediately after meal or your lunch or breakfast or dinner, you are going for the blood glucose test. So then it will be like, more than 500 sometimes. Therefore, before you test your blood glucose level, you should let the body to adjust that glucose back to the normal level, because you have to fast at least 10 to 12 hours. After fasting 10 to 12 hours, you have to give the blood sample for testing the glucose level or diabetic patients. So this also we have to previously prepare starch in the leaf, we have to remove. Otherwise, all the leaves will give the blue, black or dark purple color. So what is the purpose of keeping the plant in the dark for 48 hours? To de-starch leaves. To de-starch leaves. Or else you can write to remove starch already present in the leaves. If you write to de-starch leaves, enough. Food is produced in the leaf, transported to the plant. As what kind of material is transported? In what form the food is transported to the other areas? Through the phloem, that is called translocation, that is as sucrose. What is the answer for this? As sucrose. Sucrose is a disaccharide. What is the transporting form of carbohydrate in the plant? Sucrose. Through which plant tissue it is transported? Through the phloem tissue. What is the tissue that is transported food in the blood? So answer is phloem. Through the phloem, food is transported. That what is the single term given for this transportation of food in the plant? Translocation. What is the single term? Translocation. It occurs both upwards and downwards, or vertically, in all the direction. After 5 hours later, you are testing this for the starch. Write the steps of the starch test. First, you have to boil the leaf in hot water. So in the starch test, number 1, boil the leaf in hot water. Number 2, boil the leaf in alcohol using water bath. Okay, first one, boil the leaf in water. Boil in water. What is boil? Leaf. Boil the leaf in hot water. Why plant leaf is boiled in water? To kill the cells, and to make the to break down the cell wall and to increase the permeability of the cell membrane. Boil the leaf in water. Second one, boil the leaf in alcohol using water bath. alcohol using water bath. After that, you have to rinse the leaf with water, or rinse or wash the leaf with fresh water. Why rinsing the leaf with water? To wash away remaining alcohol. Clean water. After that, the last step, adding few drops of iodine solution. Adding few drops of iodine solution. But you have to keep the leaf on a white tile. That's better, right? Keep the leaf on a white tile. Keep the leaf on a white tile and add few drops of iodine. On a white tile and add few drops of iodine. So that's how you perform the starch test. But boil the leaf in hot water to kill the leaf, or else to make the cells non-living and to increase the permeability of the membrane. Second one, you have to boil the leaf in alcohol using water bath. Why? To remove chlorophyll. Number 3, rinse the leaf with water, or you can put that back to the initial water sample or hot water to wash away the alcohol remaining. Otherwise, alcohol and iodine will react and will give some other color. Keep the leaf on a white tile. After washing with water, you have to keep that on a white tile and add few drops of iodine. Then you can, after that, you can get the observation. So what are the observations of the A and B after this starch test? Leaf A, no change. Leaf A, the covered part, no change. Covered area, no change. Covered area, in A, covered area, no change. No color change. No color change. But in B, color change occurs, or turned blue-black. That turns into blue-black or dark blue. Write the balance chemical equation to that food production process in the leaf. That means indirectly they are asking balance chemical equation for photosynthesis. Every year oral paper you will be getting a question from photosynthesis and respiration. So you can write here 6CO2 plus 6H2O arrow C6H12O6 plus 6O2. Above this you have write light or sunlight. Below this you have write chlorophyll. Right. So photosynthesis experiment, but pay attention. Next on part B, I'll erase the screen. Part B. Below diagram of a blood vessel and few cells that make it up, name A and B. Two special features that can be seen in the tissue A. You can see here and few cells that make it up. So if you take blood vessels, so it's surrounded by epithelial cells. So this represents epithelial cells. And this is the smooth muscles. Wall of the blood vessel made of smooth muscles. And it's lined with epithelial cells, blood capillaries. Epithelial cells. Tissue A, that they are asking two special features. Cells are placed on the basement membrane. Nucleus present. Nucleus present. Cells are placed on the basement membrane. cells are on the basement membrane. Epithelial cells ne. Epithelial is like if you take the wall of your room as smooth muscles, the brick and the sand and all the interior, and the surface of your wall, the paint, can be considered as epithelial. Actually, epithelial tissues cover the inner, external and internal surfaces of your innermost surface of your mouth, innermost surface of the bladder, the stomach, uterus, vagina, mouth, buccal cavity. Right. Next one. Write two characteristics of the tissue cells that make up the heart wall that differs from the tissue cells shown in figure B. Heart. Two characteristics of the tissues are cardiac tissue, that's branch and involuntary. So one characteristic you write branch. Cardiac is, that is the only branch one. Branch fibers or the branch cells. Cardiac. The other most important in cardiac, that is striated, but smooth unstriated. If you have done this, you can mark with red pen. Cross strings are present. They are asking two characteristics. Part 4. Specify location other than the wall of the blood vessels where the tissue A found. So, tissue A found in the wall of the blood vessels as well as innermost surface or alveoli of lungs. In addition to that, kidney tubules, alveoli of lungs, you can find epithelial tissues. Buccal cavity, or mouth cavity, innermost surface of the vagina, urinary bladder, innermost surface, you can find the epithelial tissues. Some of the places, gas exchange. The other one, absorption, secretion. Even in the small intestine, villi also contain epithelial tissues. There are many other locations where you find the epithelial tissues. The small intestine, absorption of nutrients, absorptive function, the small intestine. Kidney, in the nephron filtration of blood. Alveoli of lungs, gas exchange. Next one, what tissue contributes to the transportation not mentioned here? What tissue contributes to the transportation not mentioned here? That means blood tissue, actually it's blood tissue, its transportation, but not mentioned here. So what is the tissue involved in the transportation? Blood tissue. It's a connective tissue. Main function is transportation of substances, but it's not mentioned here. Because here it's mentioned cardiac, smooth, and epithelial. Next one, question number 5. From nervous system. Brain, the main parts and the functions of each part you should know. But last few years oral papers, they haven't given much diagrams under system lesson. Mostly the students memorize labeling, drawing the diagram, kidney, lungs, nephron, nervous system, brain, digestive system, respiratory system. Mostly they label and they draw it, but very few questions they have asked last 4 years papers. Therefore, you don't need to you don't need to pay much attention about this drawing those diagrams and labeling much. Right. Next one, name the parts A, B, C, D. C will be the largest part of your brain, that is cerebrum. B, cerebellum. A, medulla. B will be cerebellum, the second part. So the largest part, cerebrum. Second largest, is cerebellum. And A will be medulla. What is D here? That is skull. Or else you can take actually not the skull, you can write meninges here. So connective tissues, meninges. Between these meninges you get a fluid filled medium. We call it cerebrospinal fluid. What is the part which controls the memory, intelligence, thinking, vision, taste, touch, pressure, all this under the control of cerebrum, that is capital C here, or you can write cerebrum. Cerebrum. Three, write down the functions of medulla. Controls involuntary actions. Medulla controls involuntary actions. Such as what you write, respiratory movements, heartbeat, vomiting, sneezing. Involuntary actions, you can write two involuntary actions. Controls the heartbeat, controls the respiratory movements. That is the function of medulla. Next one, what is the process? Withdrawal of the hand immediately when contact accident with a hot object. We call it reflex action. What is called? Reflex action. Sudden automatic response without thinking. Such actions are called reflex action. If your hand contact with hot object, immediately you withdraw hand. What is this called? Reflex action. Reflex, the functional unit of the reflex action will be reflex arc, ARC, reflex arc. It consists of three neurons, sensory, motor and intermediate neuron. Briefly explain the above process using the flow chart. So withdrawal of the hand, you have to write, first you get the pain, right, or the heat. So right here, part 5, you get heat, the pain. So who receives this? The skin. That is the receptor. From the skin or from your hand, there will be a signal sensory neuron. From sensory neuron, it's to interneuron. From interneuron, that is actually, or else you can write here CNS, central nervous system. And then motor neuron. From motor neuron, it goes to the muscles of your hand. Muscles of hand. What is the response? Contraction of the muscles. Muscles of the upper arm. Response will be, what is the response? Contraction of the muscles of the hand. Contraction of the muscles. And you withdraw the hand. So what is the order when hot object contact with your hand? Heat. Skin will be the receptor or the hand. Sensory neuron, interneuron or the relay neuron, motor neuron. Effector will be the muscles of the hand, because you are given an example. For that example you have to write it. You can't just write the receptor and the effector. You're right. What is the receptor? That is key. What is the effector? Muscles of the hand. If you feel the smell of the food, then the receptor will be in the nose. Effector will be the salivary gland, secretion of saliva. And this time all this respiratory system is very, very important. This time all which system is more important? Respiratory system. Viruses, characteristics of viruses. That's very important. So this one is not hard. I think it's almost 12. I'll quickly tell the answers. A, B, C, A, trachea. A, trachea. Right. So you can write trachea here. Then you get B will be the right lung, left lung, left lung. A will be trachea. Then you get bronchus, bronchioles. C will be bronchioles. D will be alveoli. Write down the importance of the cartilage rings here. In part A, there are cartilage rings. That is it helps to maintain the cylindrical tube-shaped structure. Maintains what? It helps to keep cylindrical tube structure. Otherwise, it will collapse. Cartilage rings helps to keep the cylindrical tube structure of the trachea. Structure D is necessary for respiration. What is the special name given for D? That is alveoli. Alveoli sac. That is important for respiration. Gas exchange take place through D. What is the process take place through the structure? That is diffusion. Diffusion of gases. Diffusion of gases occurs through the structure D, that is alveoli. And they can ask about this COVID-19 symptoms and preventive measures, and about the virus, RNA virus attack here, the respiratory system, corona, COVID-19, and the characteristics of viruses, and how to protect from viruses, other diseases caused by viruses. Either they can ask questions. And this is also very important. Adaptations of D to make the process efficient. Actually, adaptation means alveoli, the surface, the moist surface. Moist surface. That is one adaptation. Gases get easily dissolved. The alveoli surface, that is the respiratory surface, moist, wet. And the other one, thin wall. Thin wall. And the other important thing, high surface area. High surface area for the efficient gas exchange. Moist surface, thin wall, high surface area. And highly vascularized, that also get an adaptation. Alveoli surface. Respiratory surface, alveoli of lungs. Singular is alveolus. Thin wall, high surface area, highly vascularized, good blood supply. Two changes that occur, inhaled air through the nasal cavity. One is what? Moisturizing or humidifying. When you breathe air, water vapor is added, humidified. The other one is warming up of air to the body temperature. What changes occur in the inhaled air? Humidified, and in water vapor, warming up to the body temperature. Write down the function performed by the cilia of the nasal cavity. Remove dust particles and germs. It removes dust particles and germs. Cilia. Otherwise, what happens? All the dust particles will enter into your lungs and cause infection, germs. Last one, the table. Hormones. You should know the hormone, the site of production, and the function. Thyroxine produced by thyroid. What is the action? Controls metabolic rate. Metabolic rate controls by the thyroxine hormone. Number 2, ovaries. What is the hormone? Estrogen and progesterone. Estrogen and progesterone controls secondary sex characters of females. Secondary sex characters of females. Regulation of blood glucose level, hormone you can write insulin. And glucagon also, if you want, you can write here. Site of production, pancreas. Pancreas secrete insulin and glucagon, beta cells and alpha cells. Growth hormone, that is also called GH. Who secrete growth hormone? Pituitary. Pituitary. Now remember, your brain, there are two glands attached to your brain. Number 1, pituitary. Number 2, hypothalamus. Pituitary is just below hypothalamus. What are the two glands? Endocrine glands connected to your brain, associated with your brain, hypothalamus and pituitary. Pituitary is just below hypothalamus. And what is action? Controls the growth of the body. Controls growth. That means protein synthesis, cell division by growth hormone, elongation of the bones. So hormones, you should know the common characteristics also, and they are chemical messengers, chemical coordination by endocrine system, especially regulation of blood glucose level, regulation of body temperature, regulation of water level. They can ask under essay type questions. Regulation of water level, regulation of blood glucose level, and regulation of body temperature. So those three, keeping those conditions constant in the internal environment, is known as homeostasis. Therefore, anyway, you will be getting not only from one system. Question number 5 essay type, A, B, C, D. Sometimes there can be 3-4 parts. At least from 3 systems you will be getting the questions. If you don't get any questions from the systems in the essay or structure essay, definitely will get at least MCQ. Example, digestive system. If you don't get any essay type questions, at least you will get one MCQ from the digestive system. Therefore, to go for good pass, you have to go through the whole syllabus. There's no shortcuts. If someone is telling that one is coming, this one is coming, you should ask from them what is not coming. No one can tell for 100% confirm. No one can 100% confirm this one is not coming for this time all. No one can tell. Even the other subjects like that. So you will get like, that seminar, this seminar, that one, that set all, that one is coming, this set all, this one, like that. But anyway, you should ask from them what is not coming. Can you 100% confirm? No one is telling like that. Therefore, my advice, don't find shortcuts. Go through the important areas. Then you will get good results. Practice many questions possible like this. Right, now today, now is almost 12 now, midnight. Now, that's all this paper. And I think we finished both the papers today except the MCQs of this paper. And tomorrow night, same time from 8 until midnight, we will be doing the chemistry seminar. That will be the last seminar. Today is the second seminar, this seminar series. And tomorrow, Saturday night, the final chemistry seminar. So you can join with that seminar also, and we will be sending there's a separate group. And if any of your friends want to revise the grade 10 and 11 chemistry, they can join with the tomorrow's seminar. So I hope you definitely you have improved a lot today with my teaching, and Sir Channa sir also did his best. And so you can join tomorrow with chemistry seminar. Until then, take care. God bless you.